We can go down at 30.
30. Copy that. Going down. Okay, heading down at 30 meters a minute. Bring that drawer in too.
You want to disable auto heading, Don?
Yeah. Got it. I brought the drawer in too, so we're closed up now.
Actually, Don, I don't know. When I was co-pilot, I don't remember if I enabled your thrusters.
No. Really?
Serious thrusters?
Okay.
Serious thrusters.
The thrusters page.
Oh. Yeah, but we see them on here. No? Control. Oh, enabled, you said. Okay.
Yeah, just make sure I did that.
That's
D2.
D2, serious. There we go.
Yep.
Okay, and thrusters.
Okay,
no, you
punch
did
that.
not.
There we go. Bingo.
Okay.
I was looking at ground
All right,
points.
at bottom lock, 30 meters.
Copy
We
that.
can start slowing down.
Okay.
60 meters, excuse me.
Okay, I've got it at 55.
Good morning, everyone who may be joining us on this wonderful Sunday morning from the Okeanos Explorer. We are diving in a site today known as Penchant Basin. This area is thought to be geologically active. It is our dive site is located south of the largest brine pool, as well as some of the largest chemosynthetic habitats known in the Gulf of Mexico. And additionally, it's also thought to harbor a lot of coral communities, making the most of those asphalt extrusions or orthogenic carbonates.
20 meters.
So our
Copy that.
aims during this dive are going to be to climb a slope, a
Nope,
westward
squid,
face,
yeah.
well, head due west, climb a slope, and from there run along a topograph, well, a
15 meters.
bathymetrically high ridge to the south and then
Nothing in
in
sonar
a southeasterly
from me.
direction.
Sonar,
We
no
do
bottom in
hope
sight
we'll
either.
we'll touch down an area
it'll
where
come up. It
we'll
might be
see
kinda
some
hazy.
kind of chemosynthetic habitats, as well as, I can just see the bottom looming in sight, as
Yep,
well
here's
as
bottom.
Okay,
then
copy that.
potentially have some corals in the areas of higher terrain. Of course, these potential chemosynthetic habitats are associated with oil and gas, and that means that we are dodging a number of
He's got an
hazards.
altitude of
For
16. You want him to
instance,
stop?
Okay, you
just
wanna
south
stop?
of us,
Yep.
there is a absolutely humongous drill ship that
All right, if you want to engage auto heading,
Okay.
means that has a
come to
2-mile
starboard,
exclusion zone
like 30
around
degrees.
Copy
and
that.
so our dive track is has been forced to be
225.
a little bit more north.
225. Copy
So
that.
we are hoping, despite that, we do see an exciting array of animals and habitats today, and should be coming off bottom around 4 local, which is Central Standard Time today. We've just touched down on bottom in what appears to be a sedimented area.
Okay.
A
I'm
couple
on
of fish and
225.
lots of jellies in the view. So while our pilots are orientating themselves and just getting set
Don,
up,
if you want to
we'd like to take this opportunity to
bring my-
introduce
Set your
ourselves
arms
so you
up.
know who
Well,
it is you
Oh,
are
you're not yet.
Bring the
listening
wings
to today.
outboard.
So my name is
Copy
Diva
that.
Amon, and I am a research fellow at the Natural History Museum in London. There I work on chemosynthetic habitats such as those we hope to see today, as well as human impacts on the animals and life within our deep oceans.
And this is Chuck Messing.
Copy that, Bridge. Thank you.
On the faculty in the Department of Marine and Atmospheric Sciences at Nova Southeastern University in Fort Lauderdale, and I'm an invertebrate zoologist. My particular area of
Wings are outboard.
specialization is the
Camera's
crinoids
looking
seen in
straight
visible
down.
stars, but I also have a fondness and some experience with deep sea coral and other hard-bottom communities.
And I'm Alex Avila.
You wanna
I am
come
a
up a little
PhD
bit?
in fisheries at
Copy
Oregon
that.
State
That'd be good,
University
'cause I'm
and a Dr.
gonna head
Nancy
towards
Foster
you.
Scholar,
Okay.
which is part of the Office of National Marine Sanctuaries, as well as an Oregon Sea Grant Fellow, and I'm here
Come
to
up
help
20
out
meters
with
off the
data
bottom,
logging
good?
and
Sure.
help
Yeah.
process samples.
Bridge RV nav. Let's set bail out for northwest for now.
Okay, we're 20 off.
Thanks, Bridge.
We need a little bit more time. We're setting up right now. So.
So one of the best things about the Okeanos Explorer is that not only are you listening to Chuck and I and Alex today, we are actually joined remotely by a range of scientists with various
Brian
expertise
probably, huh?
who are located around the world. So we stream
Here I am.
all of
Okay.
our
If you want to tilt up.
Coming up.
imagery
Yep.
as well as data live, and that means that folks ashore can
Sure.
actually have access to this within 2 or 3 seconds in some
Gonna
cases.
come down a little bit.
And so that means that while we may not have the room
Okay.
on the ship to house
Let's get
all of
this
this
guy
knowledge
real
and
quick
all of these
rolling.
individuals,
Let me get
we
your
can connect
uppers
with
and lower
them,
swing arms
Yeah,
up.
make sure they're
via
all the
telepresence.
lower
Yeah,
ones. Make sure they're
and
outboard.
Yeah,
they
outboard,
can
yeah,
help
copy
to
that, yeah.
influence this dive by giving us their expertise.
And deploy. You want them all the way or half?
Just partial.
Partial, just like that. Okay.
So
Upper
today,
swing
I'm
arm
not sure who
coming
we have
out
on the line.
starboard
Do we have
side.
anyone joining us on the line? It is Sunday morning, so perhaps not.
Hi, this is Marissa Nuttall. I'm a researcher at the Farnham Bank National Marine Sanctuary. The site we're on today is just outside of one of the proposed areas of expansion for the Farnham Bank, and so we're here to try and look at the connectivity
Outboard.
between the sites.
Nice.
Great, thank you. Anyone else on the line
Deploy.
today?
This is Cody Kramer from BOEM. I'm a geologist.
Okay. Upper.
And Carolyn Ripple, Marine Geophysicist with the U.S. Geological Survey outside Boston.
Okay. The lights are up.
Yeah, copy that.
I thought we had Bill Shedd on the line as well. He may have nicked off.
I think it's drifting away from you fairly fast.
Yeah, there's quite a bit of current
Well,
down here.
thank you for
Right,
all
coming
for
out.
joining us on the phone line. That's an extra bit of
jelly below you.
commitment sometimes, but we also are joined by a number of scientists who connect with us via an instant messaging service. And so today we're joined by Asako Matsumoto, an
Tyler's
Iberian
kind of blowing your
expert from
face down.
Japan.
It's kind of annoying.
Carolyn Ruppel, who you just
That'll
heard
come
from.
up a little bit.
Mark Benfield, who has quite a lot of experience working in the Gulf of Mexico.
I'm going to have to look that one up. That looks like I'm going to throw out a guess here and say Hybocodon
I was going to say benzocodon.
or Benzocodon. Yes, something codon.
Very
And,
close
we'll
to
look
you.
that up.
Yeah.
Get back to you. That's a hydrozoan medusa.
Squid at the bottom.
We're also joined by Megan Cromwell, Megan McCullough, Bob Carney, Scott France, who is has quite an important role in. Wow, okay, there are lots of clamshells on the floor. Sorry, Scott. So Scott France, who is from the University of Louisiana Lafayette, and who has quite an important role in the next year of expeditions and potentially on from that. Which will be in the Atlantic. And last but not least, Tara Luke Hammer has also joined us. So the Okeanos Explorer is exploring the Gulf of Mexico during this expedition. We have 4 more dives, including this one.
Yeah, we can.
For
Get moving
this
now if you want.
EX1711
Okay.
expedition, which is quite sad.
So you
But
got Cirrus lined up at
then
210. I got
we,
waypoint
in
1
March,
at.
I
will
just moved to
be
just get
taking
that
part
out
in
of my
a,
face directly.
Oh, okay.
mapping
All right.
cruise
So you guys
to
Fine, Enroll.
gather more
230,
data
am I
from
right?
the
You
Gulf.
guys?
Sure.
And then finally, in April next year, there'll be
Are
a
we
second
heading
RV
towards
cruise in the
something?
Gulf
Just a
of Mexico.
waypoint.
Just
This is
waypoint.
the first time in 3 years that the Okeanos Explorer has been back
Lasers are
in
clear.
this
You guys
area.
good with 30?
Copy
For
that.
the last
30
3 years,
meters?
they were working on
Should
the
be fine.
Capstone
Sure.
project, which took them to all the different corners of the Pacific Ocean.
Bridge RV now.
After that, they should be heading up the east
Yeah, I'd like to
coast
move
of the U.S. to
range
investigate
30
some deep
meters,
sea
bearing
habitats
230
there.
degrees,
So with
speed
that,
0.2
we're hoping
knots.
for a really great dive today. I already see some signs of chemosynthetic life. So let's get on with it.
Diva, do you know what that gas
It's a good
pod
copy.
is by any chance?
Give me a second.
If it's associated with the clams, it might be associated with a
Partial.
the seep community, and that I believe is a synaphobranchid eel. Is that correct?
Uh,
Or not? I'm on
debatable.
thin ice with my fishes, and you're right. That jelly was benzocodone. Hydrocodone is apparently a pharmaceutical product of some sort.
Move initiated. Thanks, Rich.
So what we're looking at here are definitely some clam shells. So clams are one of the main inhabitants of
this?
chemosynthetic
That
habitats
sure looks like
here
a
in the Gulf
brine
of
pool.
Mexico.
Yeah.
These are all dead, but the clams utilize sulfide-rich chemicals
Did she say
seeping
we've
out
been,
of the
or
seafloor,
they dove it before?
No, they
using
said, like,
their
north of
foot,
here.
Come on
which
out.
Oh, okay.
tends
I'm
to
not
be,
sure how far north.
buried
Well,
down
there
in
was
the
a lot of
sediment.
There is one that
You
I
can
used
also
to
see some
dive a
kind
lot.
of ophidiid just above
It's
us.
big, like 100 feet long.
As well as that gastropod,
70
which
feet
is
wide.
seep-related,
Okay.
definitely. I'm just trying to find out what it is.
Let's get
So
this
we're
fish.
obviously in some kind of geologically active area where there is likely seepage coming to the surface of these hydrocarbon-rich fluids, and that is what powers life here at these sites.
And is this a bicytid, I think, or an ophidiid? You can see it's a couple of little, very, very slender little chin barbels that it uses to probe the sediment.
Going to change my heading a few
Yeah, come on
degrees to
out.
keep you in view.
Direction of movement was 230.
Copy
Just
that.
letting you guys know.
And there's a little hatchet fish.
I think that might be Barothrites pari, maybe?
Okay. Little hatchet fish is in the family Sternoptichidae. They are midwater fishes, and as we saw yesterday with some viper fishes, these are not specifically bottom-associated fishes, that little deep-bodied fish. And there's another midwater fish that's probably a myctophid, a lanternfish. So these are midwater fishes that migrate vertically, sometimes hundreds and hundreds of meters every day up towards shallower water at dusk and down towards deeper water at dawn. And here at our depth of 800
do a snap here.
meters, 804 meters, they've just run into bottom, whereas they normally might find
Right,
deeper
come on
water.
up.
Not sure what that was. And
It did look like that.
okay. And so by the time they migrate up again, overnight the current might sweep them out to where the next day they can find deeper water when they migrate down again. There's only scattered bioturbation here. I can see a number of trackways left by deposit feeders, probably sea cucumbers or perhaps sea urchins.
Let's get this guy in the middle. If we can.
There's an planktonic arrowworm, a ketognath, which is a major planktonic predator. They have a head full of curved spines with which they grab prey.
Okay, coming out. A lot of current
And
today.
lining their bodies, they have a series of little, extremely small hair-like fans.
Come on in. Yeah, there's
that
quite a
they
bit of
use
current.
to, sense,
Still getting a sense of it, but it seems to be
vibration sensors.
blowing me sideways.
That's the primary
Yeah.
sensory world is vibration.
Come on in.
And I think we've seen this one before too, but I don't remember the name. It's got those very, very long lower pectoral fin rays that again act as feelers in the sediment, and the ones spread out perhaps as antennae as well. Dicrolini. Dicrolini, I'll get it. Intronigra. Dicrolini intronigra. You can see the extremely well-defined lateral line just underneath its long dorsal fin.
Hey, pilot, we're
and
looking
the
straight
pores
down
on
on
its
you.
head,
Ship is
which
about
are
done.
extensions of its vibration.
Video is
sensing
clear.
Don't know the move that is.
Okeanos receptive lateral line system. And our laser dots are 10 centimeters apart.
Move complete.
4 inches.
Thanks,
so
Bridge.
that fish was only
Yeah,
about
pods.
5
Moves
inches
complete on the
long.
Okeanos is done
And
moving.
here we're seeing
Shouldn't swing for too long because we're pretty shallow.
white
So.
bacterial mats, which are indicative of chemosynthetic activity. The little black patches are anoxic sediments. What have we here?
Let's get this guy in the center.
Saw one of these a couple of days ago. It's this pagurid hermit crab that has an... Actually, this one does not look like an enemy. This looks like the one that has the colonial... Oh, zoanthid.
Oh yes, that's exactly right. Good eye, Diva. And there are the 1, 2, 3, 4, 5 polyps. I guess this hermit is camera shy.
I just turned those off.
So
Doc.
funny, I remember being taught about this during my first year of university, which was a long time ago,
All right,
in the
you
marine
need to get on
invertebrates
your way.
class. But I'm just trying to find, Epizoanthus. I think
Just trying to
is
run away
the,
from you.
Yeah.
the
Well,
genus.
that makes sense. And very long legs for a hermit crab.
Let's get the fish real quick as I come by.
So this does look like a snaphorhynchid, but not obviously the usual ones we're accustomed to dealing with. This one could be Iliopus,
get the head.
was it?
He's got protruding nostrils.
Right.
Yeah, Iliopus, I think.
There's quite a bit more bacterial mat here. Bright red shrimp.
Snap on the fish.
And narrow trails here. That looks a bit different. Shorter snout. Nostrils are not out at the tip. Doesn't appear to be.
As we're zooming out, we can see a Nematocrustinus shrimp and lots of these clam shells, likely vesicomyid clams. Ooh!
No, it's wide open.
What is that?
A squat
I'll
lobster.
close it here.
Yep.
Definitely just shot out of the
No,
sea
I
floor.
don't think
Oh
that's
no,
a squat
it's
lobster.
Come on in.
Wait, that's a
Oh, it begins with a P. Polychelid.
Polycheles.
Sure.
That's a lobster-like creature. He's got very long, slender claws that are tucked in along the body here. And this, you might also say, belongs to a group of invertebrates that could be called living fossils, because the group, and there's a rat tail in the background. This group was quite diverse, I understand, during the Mesozoic, and there is a
tripod fish.
A tripod fish in the background, along with another eel. That's the first Polycheles that we've seen.
Go ahead and drift down to the tripod. This guy's very different than the front.
And Bathypterois is the tripod fish.
Lasers on real quick.
Patterning on that one.
Okay, laser's clear.
Don't mess with that guy.
Yeah.
Oh, and he's got a really big parasite on
Oh,
him.
it is hanging down from
Let's get the full body.
the part of his pectoral fin.
And
His or her
as you
pectoral
come
fin.
in, if we can,
Very,
once we
very
get at this,
tiny
if we can
eyes.
rotate around the right,
And resting on the
go around his
elongated
front side.
rays of its lower caudal fin and pelvic fins, with its elongated rays of its pectoral fins, very long and slender and used effectively as antennae. You always see these fishes sitting
you
nose
can go ahead and
into
pirouette around the
ground.
right now.
These are also known to be ambush predators, so they sit very, very still on the sediment surface, and when some possible food seems to drift by, it will suddenly grab it and have a nice little munch. We can see another one of those gastropods just to the left, which you can see the siphon extended from, indicating that it is alive and munching along the seafloor there. It could be a Phymonorhynchus,
You can
but
go head in if you want, Roland.
I'm not sure. Oh, this one has 2 parasites on it.
Oh yes, look at that one on the other fin. What are those?
Where did... Oh.
Can we get a zoom in on that?
This is a good angle shot if you can sit. Max zoom.
Alright, I'll get closer. Coming out.
Full wide.
So in the deep sea, there are a number of parasites that live on
Well, we're going to spend a
deep sea
second
fishes.
here.
These can be, well, not just deep sea fishes, lots of deep sea animals, but mostly fishes. They can be anything from barnacles to amphipods to isopods and even copepods. So mostly all arthropods.
There's somebody slowly
Alright, go ahead and roll
along the bottom.
Tilt down.
Try not to
Background.
dust it, but we might get a little dust. Go ahead.
And this may be, oh, look at that. That's
That looks like an amphipod to me.
yeah.
Yep, so the top one is definitely...
And this is probably Bathipterois viridensis.
So they both look like either amphipods or isopods.
Well, the lower one looks more like an isopod. I can't see the upper one. I didn't know there were amphipod parasites of fishes. That's interesting. Very cool.
Oh, it has 3.
Yep, there's a little one behind the right pectoral fin.
So these are likely impacting this fish's life to some degree. They'll be either, drinking its blood directly, or perhaps benefiting from some of the food it may be eating. But regardless, they will be impacting it in such a way that makes the animal itself, this fish, have to work a little bit harder and direct energy away from its own growth or its own reproduction to replacing whatever it is these parasites seem to be consuming.
Yeah, when you're good, Roland.
Okay, I'll call a move in if you guys are ready.
Sure.
17. Bridge driver nav. Yeah, I'd like to
Get
move
this,
range 30
fish real
meters, bearing 230 degrees, speed 0.2 knots.
Go ahead in.
There's a rat tail.
Good copy. Thank you, Rich.
Acroridae. Have we seen this one before? It's got a
Perhaps,
little
I'm not-
parasite on its anal fin there, a little white comma-shaped creature. Looks like an amphipod. Little chin barbel there, covered with mud.
Wow, this one's eyes are
Very buggy-eyed. So we are still at a
There's another
depth
parasite down there
where light in the
under the fin.
open ocean can reach the bottom, even though it's very dim. So some of the fishes here will have very large eyes.
Well,
like
there's parasites all over that guy.
the tripod fish,
Coming
of
up.
course, doesn't. It's relying on the sensitivity of its fin rays to detect its prey.
Crab
and
ahead.
danger.
Left of laser.
light can get to the seafloor at 800 meters.
Well, the
Go ahead
mesopelagic
in.
zone goes down to 1,000 meters, and that's what I've been told anyway, that it can be detected that deep. That's an interesting crab, a little spider crab of some sort. Looks like a mithrax or mithracid, but I can't quite tell. I'm not sure of my spider crabs.
Video's clear.
Yeah, coming up.
As this cruise has gone on, and I've seen more and more things that I've never seen before, I've become less and less certain of commenting on things.
Large Holothurian.
The fishes, it goes without saying.
So during this expedition, we've seen quite
Go
a
ahead.
lot of Holothurians, quite a large diversity of them.
And there's one right now.
Quite a lot of in purple. I'm just trying to find out which species it is. It's got these very tiny papillae on its dorsal surface, as well as this very obvious striated fringe. Perhaps it could be Benthodites sanguinolenta or typica. Those are pretty difficult to tell apart without specimens in hand.
Look at those very, very fine little filaments coming off its dorsal surface. That's wonderful video. Look at that.
And usually these have polychaete, which lives alongside it and can actually shelter underneath its fringe or with it, under the underside of the body. I don't tend to see the one here, but
it looks like it's run up against a little stalk and perhaps wondering, oh, where do I go next?
Video is clear.
Copy that.
So the old worm to
Lasers clear.
Ooh! So sea cucumbers
Jet
can
propel.
swim if pushed. Some species do swim more than others. For instance, there's Pelagothuria, which is completely pelagic. And then, of course, there's Aneupniastes eximia, which we've seen quite a couple of during this expedition. And those do spend a lot of time swimming, though are not strictly pelagic. This one likely uses its swimming abilities as an escape mechanism, and so will only do so when really pushed. And I guess the presence of D2 was a little bit too close for comfort. There's a chimera just approaching to the right.
Oh, yes.
So these are known as rabbit fishes or spook fishes.
Go right in.
This will be our second one, I think, for the expedition. The one we saw before was a long-nosed one. You can see just on the front of the dorsal fin, it's got a venomous spine, as well as you can see some very pronounced sensory organs on the front of its body. I've never seen one dead on like this, and this one seems to have a really weird proportion of its fin size compared to the rest of its body.
Yep, he sure
This
is.
is definitely a different species.
He's very curious.
Very close right now.
Oh, yeah.
Oh, this one's got a very blunt nose. This is not a species I've seen before. But I don't have a lot of experience with this.
Look at how long the tail is.
Hopefully this one will be not difficult to identify by chimera experts. It's very strangely proportioned. Do we have any sense of how big that was, Pilot?
Do we have lasers on?
No. Lasers are on now.
I would guess it's quite small, actually.
Yeah, it's pretty small.
Probably, if you include the tail, about 25 centimeters, maybe?
Yeah, less than a foot in length.
Oh, yeah.
Copy that.
Oh, wonderful.
Yeah, I turned
You can
the port
see
a
how
lot
much
to
of its
keep it
body
in
is
view,
actually tail compared to head and
Kill lasers.
Copy that.
main body.
That would make
Brilliant.
a great cartoon character. It's already got big enough eyes.
So, Dave, I'm looking at the FAO guide, and it looks like it's a Chimera cubana, possibly.
Great.
But that's never been reported from the Gulf of Mexico before.
Well,
Cuba.
it's close.
What about Hydrolagus alberti?
The spines are a little bit different, but it could be that one too. Maybe I was looking at the shape of the snout, actually.
Yeah, look at the shape of the dorsal fin is rounder than
Let's let him go.
Yep. You are
I'm
correct.
going to get back on heading here. I'm going to come back
We've
on.
been working on working with a number of ichthyologists ashore, one of them being Ken Sulak, Kevin Rodebacher as well, Tracy Sutton, and then the last chimera that we saw, which was a Harriota species, a long-nosed chimera. I sent that off to a chimera expert, Brittany Finucci, and she was very helpful in identifying that as well. So lots of people that we'll make sure and get those images onto. These are very rarely seen alive most of the time. I remember when I sent the last pictures to Britt, she was saying that this was the first she'd dissected, hundreds of that species, but she'd never actually seen one alive. So This is why we do exploration, and they're able to get these images that we do.
Okay, back on heading 230.
So it looks like we've moved out of that area where we saw a lot of those shells and some of that reducing
Do a quick
reduced
zoom
sediment.
here.
That looks like a glyphocrangon
Yes,
shrimp.
it does.
And we can tell that because of those really characteristic big eyes and that stubby little body with quite a thick-looking, robust carapace.
Alright. Push out.
Okay.
Let's get this fish real quick, Roland.
We've already seen quite a couple things that we haven't seen yet for this expedition, so that's quite exciting.
All right.
Coming tight.
What do you think this one is?
This looks like some kind of midwater fish,
Argentine?
which I'm not very good at.
Yeah, I don't know. I'm looking for chimeras with rounded dorsal fins. Haven't found one yet.
Alright, coming up.
Jelly in the water.
Go ahead in.
And here is another one of this, one of these hydrozoan medusae. This is different
That's
from the
different.
other one, yes.
So are we inside your lens?
And now I'm trying to back up without blowing them away.
That's beautiful.
It's drifting away
Alright.
and blowing
Coming up.
away.
Yeah, the stuff in the water column is going to come at you really fast, Roland. Hit a stick, quick zoom.
So yeah, this is some kind of terrestrial plant matter,
Right, coming up.
probably washed out to sea and then sank. And that's going to provide food, shelter, and substrate for
There's
a
a
variety
whip
of
coming
organisms.
left of fish.
It's coming in. I don't see the whip, Roland.
Top of screen.
Oh,
Doesn't like our lights. Come on in.
this looks like some kind of dead stalk, perhaps from a coral or a tube worm.
Tube worm, I think.
Yeah, I think that.
See the guy inside?
And he looks like there's someone inside.
Yeah, I don't think that's
One
dead.
of your chemosynthetic tube worms.
We should laser this guy. He's pretty
So this
big.
might be Cibaglinum,
Lasers on.
maybe. We can't see any plume,
Okay.
so we aren't sure. Can't be sure.
Okay. You good, Roland? Little hermit crab, we can get that guy.
And a little hermit crab.
With an actual shell, which is not what we've been seeing under most of our dives. Whereas that is what is the norm, much shallower.
Okay.
And our depth here is
Something big just swam over the top of your lights. Looks like a school of squid.
I'm not seeing that in Sirius.
I'm seeing the shadows.
You can see the scattered mounds and small pale areas.
Looks like this
of
umbilical
the
wants to put a
seafloor
turn in
here, all
itself there.
indicative
Yeah.
of hidden life, the infauna, organisms that burrow in the
I can try
subsurface.
turning the other way, putting my half turn in the opposite direction.
They could be sea cucumbers, they could be bivalves, worms, crustaceans, quite a diversity of organisms.
Yeah. Next time he's on top of me, Sirius is on top of me, I'll do a turn. See if that helps, and maybe that's what we just need to do every day. Yeah.
Sure.
Pretty
So,
driving out.
just a reminder, today we are at Penchant Basin.
Yeah, they can
at a
move
depth
30
of about
meters,
800
bearing
meters.
230 degrees,
We are
speed
working our
0.2
way
knots.
towards our first waypoint, which we landed some distance away from. But once we get there, we're then going to start to ascend
It's a good copy.
a ridge. We did expect that this low we're working in currently would have
Coming up.
some kind of chemosynthetic activity, and we have seen some of that already from that Sibaglinid tube worm, which we just saw, to the dead clams, and also the patches of
Video
darkened
swapping out.
sediment and white bacterial mats. Not a huge amount of activity, but indications that there was once much more in this area, and perhaps now it's waning.
Those floats
So
are too
Ken,
far apart.
one of our
Possibly.
ichthyologists who has been contributing during the dives, has said that he's scrolled back from the beginning of the dive and made a list of all the different animals we've been seeing. So the first congrid we saw was maybe Bathyuroconga. Then the Ophidiid was perhaps Monometopus. After that, we saw a very dark fish with elongated fin rays, and that may have been a Neobithytidae, which probably was from the species Dicroline canazawi. We also saw some Synaphobranchids,
Thought that might be a sea
finis.
star.
That's what the shape is
Tripod
there.
fish was Bathypterois viridensis, which we thought. The macrourid, we weren't sure. Ken is saying definitely a Coryphonoides, but maybe Mexicanus, maybe not. We didn't see that black ring around the eye, which is very characteristic of that species. The Chimera is a juvenile Hydrolagus alberti, so perhaps that is the reason for its very strange proportions. And he also noted the large parasitic copepods. So I was incorrect. They were not isopods. They were in fact copepods, which we saw on the abdomen and the neck in inverted commas. And so basically those burrow into the muscles of the fish and aim for the dorsal aorta. I mean, they get straight in there and go to the good stuff. And they anchor in, and from there they can suck the blood. So they are true parasites in that respect. So Carolyn Rupel suspects that we've landed just at the edge of the area, potentially with chemosynthetic activity.
Come on in.
And that's why we were seeing those couples of signs, but now we've traversed west out of that area.
Coming out.
And that looks like one of those hippoids again, not certain. One of the relatives of the squat lobsters and hermit crabs. Mostly buried, eyes up in the sediment.
Come on in.
And a clam. Yes, with a little possible anemone polyp adjacent to it. Marsh Youngblood mentioned that that other jelly we saw, the
Come on
second
in
one,
on the jelly.
and this one as well, would be also Benthocodon. Probably a different species. That's absolutely gorgeous. Like some of the other jellies we've seen, it appears to have an dark opaque red internal gastrovascular cavity, the interior space that serves both in digestion and circulation. And the idea of its being opaque has been suggested
Get the big
that
shrimp.
since it may feed on bioluminescent prey, the flashing, oh, look, we've seen this one before. This is different than the regular nematocarcinus we see. Look at those incredibly long legs.
Slightly in the same family, Nematocarcinidae, but what genus it is and species, we're not sure.
And so the jelly has this opaque gut, so that if any of its bioluminescent prey start flashing, they can't attract some other potential predator that might eat the jelly. And you can, if you look closely, you can see that most of those very long legs have tiny little claws at the tip. I think perhaps, let's see if I can figure this out. Yeah, the first 3 pairs, or was it actually 4? Yep, there, reach down and grab something. Several of its extremely long legs end in tiny little claws, except certainly for the rearmost.
All right, when you're good video, it's going to get
Ken
dusted a
mentioned
little bit.
also that that silvery fish that we saw some time ago was a member of the family Argentinidae, and the species Argentina striata. They will actually
Yeah,
sit
Don,
on the
they were
bottom,
saying that
even
we're
though they're
close
rather
to
herring-like,
the biggest brine pool in the Gulf,
or they'll drift just above the bottom.
which I'm sure
as
you've
we
dove
saw.
on before.
Yeah, we
And
did a
here
lot
we
of
are
work
coming
around.
into some more anoxic
So a little
sediment
bit
and
shallower
bacterial
water,
matter.
2,200 feet, I believe. So,
Yeah,
Pilot, can we zoom a bit here, please?
sure.
so
Come
that
on
would
in
be
video.
north of us, yeah. I'm going to turn my heading to keep you in view a little bit
Copy.
while you do this.
Not much obvious life here apart from these white bacterial mats, but there are likely many macrofaunal, infaunal species. So those are the smaller animals that live within the sediment in this area that perhaps do differ from those in areas which are inactive. These will likely have a set of fauna adapted to make the most of these reducing conditions, especially as those will likely be stronger within the sediment in this area.
And our colleague ashore, Marsh Youngblood, who's communicating via email, said one of the little medusae that we saw earlier about 10 minutes ago may be Benthocodon rufobrunia. He says it's difficult to tell. Of course, in many cases, we do need a specimen to make identification certain. And he also said, which is really interesting, I didn't know this, that we should keep an eye out on the sea floor for more individuals, because these medusae often sit on this fluff layer of organic material, this tan layer that covers the sediment, and as well as drifting.
I'm going to
just
start
above
working
the
back
sea
around,
floor
get
as
on
we've
heading.
seen.
Copy.
So what
come
you're
to
seeing here,
starboard.
that tan material is largely organic, drifted down from the seas, the upper layers above, and differs from the less organic sediment that the burrowing organisms kick up, and you can see the paler piles, the mounds of sediment that they have kicked up from underneath the surface. And it's that flocculent tan material on the sediment surface that a lot of these deposit feeders, such as the sea cucumbers and sea urchins, will feed on, or the heart urchins. I didn't realize it was that time.
So here it looks like we've got a lot more of these
Sounds good to me.
mounds and burrows, and these could be either biologically mediated or perhaps
Come on in.
geologically because of all
Bridge,
the
RV
activity
nav.
in the area.
Yeah, I'd like a move range 30
the-
meters, bearing 230
burrow looks
degrees,
like it has an
speed
entrance
0.2
and so on.
knots.
It's going to lead me to think that these
Come on out.
are as a result of some animals
Good copy.
creating homes, especially as that one has 2 or possibly even 3 exits. This should be a good depth range for us to see some of the Bathynomus giant isopods, and they do tend to live in burrows.
Move initiated. Thank you, Bridge.
Yeah, these are definitely biologically made burrows.
Let's get this dot right here. Come on
Yeah,
in.
can we zoom on these 2 black dots? I'm amazed at how many jellies, in inverted commas, we've been seeing for this dive so far. When we touched down, they were just everywhere, and I think this is the 3rd species we've seen, and definitely we've seen more than probably 10 individuals so far in total. The second one does have its feeding tentacles extended, and it's moving through the water column quite motionless, and that's likely because it's in an ambush mode, and so if it were to touch anything or pick up anything, it would suddenly grab them up, and that would be its meal for the day. But here it's staying very, very, very still with all of its tentacles extended.
Alright. Come on up.
Thank you, pilot. That was great. Another one of the Bathypterois viridensis, I think it was, in the background.
Tripod fish.
We saw one earlier.
Off to the left.
Yeah, we saw one earlier.
Yeah.
Thank you.
Oh, what's that guy? Another
Ooh, one of the jellies is on the floor.
Come on in.
Pretty big one, too, right?
Ooh, so
Yeah.
this looks like a periphylla. One of the easiest names to remember, periphylla, periphylla.
I'm going to come in
And
closer.
Yes, and this occurs in all the world's oceans.
So you kind of crash landed there.
Yeah,
And,
really.
I'm not really sure what he's doing on the seafloor. If this is normal behavior or not,
So he just kind of push himself into the ground.
or it looks like it's
Man,
having
he's,
some kind of panic attack.
he's totally out. Burrowing jellyfish.
These are obviously typically seen in the water column. That dark red color means that they are particularly well camouflaged at these depths, because red is obviously the first wavelength of color to disappear with decreasing light, and so that means it's really, really difficult to see this species.
This
They're
guy's going,
commonly
"This water
known
is
as
hard."
the helmet jellyfish, and they are found in the order Coronate.
Got a little turned around, I think.
Yeah.
I'm not sure if there is like bent knowledge of these doing stuff on the seafloor. They're found in depths up to 7 kilometers, which is pretty amazing. And it's one of these jellies that does migrate up towards the sea surface, towards its food, which of course are the plankton that will be the major organisms taking part in that migration. And so when it's full, has its feed up at the surface in the night, it will then move back down into the depths. And they are one of the most bioluminescent organisms. They emit red flashes of light, and those can act as a form of communication between each other, as well as potentially a distraction to avoid
Come in
predation,
tight. Do you have any more?
and so on.
Okay.
It's really cool how it opens and
Oh,
closes.
he's going to get
its-
tired
its-
doing that.
I guess mantle, it kind of looks like an iris opening and closing of the camera.
Yeah, and I'm not sure if it's... I mean, it's obviously trying to propel itself, but why try to propel yourself into the sediment? Anyway, interesting. We're happy to move on when you are, but video, get what you need before heading off.
That's pretty
Making
cool.
He's making progress, so he's probably coming out of it now.
All right. Come on out.
Yeah.
Just making a little trail.
Yeah,
Yeah.
doesn't look too good, not
No.
gonna lie.
Okay. We are looking down on you.
So Scott France has contributed and said that it's interesting to see all of these jellies together, and perhaps this would be a good time for them to spawn.
Move complete.
And
Thanks,
so they
Rich.
it would be beneficial for them to be in the same area as other individuals, because they release their sperm and eggs into the water column, so that having a mixture of individuals doing that ensures a good mix of genes. Okay, we're seeing quite a couple of these burrows associated with these mound complexes. That parafill is just still on the floor. Yeah, and you can see these burrows are more, some are more conical than others. Maybe different organisms, maybe an example of how new they are, how newly created they are. What's that just below? Yeah, that little pink thing.
Yep, come on in. Is it a worm?
So this looks like a
Siphon.
siphonophore, but again, why is it lying on the sea floor? Oh, and there's a polynoid. Also doesn't look particularly happy. Okay, so I may be totally wrong here, but something that I've experienced in the Southern Ocean was we were examining an area where there were cold seeps, and obviously there was obviously some kind of toxic combination of gases, and you would see squid swim into this area and then just full on collapse in front of you and just drift to the sea floor and then die. And the area was just littered with carcasses of animals that had obviously swum into the area unaware of whatever it was in the water column, and then they would have died. So maybe this is a particularly active area. In some way we're not necessarily being able to detect, and it's resulting in the death of some of the animals in the water column. That's definitely going out on a limb, though. But I do think it's a bit strange that we've seen Paraphila and a siphonophore lying on the sea floor. It would be interesting to see if we see any other dead animals in the area. I'm not sure if that's been recorded elsewhere in the Gulf of Mexico. I know that obviously brine lakes, these extremely concentrated saline areas, do cause many animals, if they were to drift into them, to die. So, but those are very obvious. It's very easy to see the brine, so I'm not sure that's what's happening here necessarily. And yeah, Scott France agrees with me. He does think that looked like a dead siphonophore. Interesting. Really interesting.
No, there's one of those jellies in the water column.
Whereabouts?
In Sirius's view.
Oh. So if you switch over to camera 2, which is the view from Sirius,
Can
that's
you
the second
zoom in
vehicle
on
that we
Sirius
have
a
on the
little bit there,
seafloor.
Bob?
That actually looks down onto D2 and gives a really
See if
great
we can-
wider perspective. But you can see one of the Paraphila in that camera. This one looking much more alive and well. The only other thing I could think of is, do these jellies spawn and then die? I don't think so. But that would explain the high diversity of jellies we've seen, as well as
Go back wide.
why we've been seeing a couple of animals that were dead. What's this dead ahead? Is that just bacterial
Snap in.
mats? I'm just on now I'm on the hunt for dead animals, basically, to try and prove my theory. Okay, nope, that's just an area of anoxic sediments, which are the blackened areas where the iron changes chemical state and results in this black precipitate. And then bacterial mats, which thrive, that particular type of bacteria thrives in these anoxic areas.
Coming out.
You can also see a glyphocrangan shrimp in that area. Looks relatively healthy.
Snap in here. Yep.
As well as another one of these hermit crabs with the epizoanthid living on, instead of its shell, a colonial zoanthid. And then another one of these tube worms. And those tube worms are very indicative of chemosynthetic
Bridge,
activity.
RV-nav.
But if you were tuning in yesterday, you would
We
have
had like a
seen
move,
we
same as
sampled
last,
one,
range
in that
30
same family,
meters,
a
bearing
cingulate,
230
and
degrees, speed 0.2 knots.
what was below the sediment was probably 2 or 3 times the length of what we were seeing
It's a good copy.
above the sediment. So these animals can extend very far down to the sediment, and there they can mine the sulfide-rich fluids, tap into those, and use them as a source of energy. So even though we may not be seeing much of this activity on the surface of the sediment, it may be very, very active further down in the sediment. It looks like another one of those gastropods, but this one is dead.
Snap on the fish.
I'm not sure. We've seen one of these already for the dive, so
Okay.
Ken would have identified it, but I'm not sure which one in his long list that would have been. Perhaps the dicrolian? No, that's the one with the elongate fin rays.
Let's get this guy. Come on
Perhaps
in.
the monometopus. And this is another synaphobranchid, which is an eel-like fish. That one could be Synaphobranchus affinis, which we've also seen one of during this dive so far. And here you can see the lateral line extending down the length of its body. And so that is one of this fish's key sensory organs
Coming up.
that it uses to detect vibrations and in the water column. It uses it to detect movement of other animals, vibrations, pressure gradients, and so on. And so that's kind of like our nose or
Snapping
our
on this guy.
the hairs on our arms. So this is a macro rid. We haven't seen one of these for this dive so far. Maybe a Nizumia or something, but this one has a very characteristic looking tail. Interesting. And you can see it's almost in its little pit it's sort of excavated. Some really interesting observations for this dive so far.
Alright, when you're good.
Another one of the Nematocarcinus shrimp, those long-legged shrimp we've been seeing throughout the expedition. And now it seems like the jellies have moved away. We're moving slightly. Actually, no, we're not moving upslope yet. We are sort of, moving across a gently sloping area, and that slope should build as the dive progresses, and we traverse up to a local high, a ridge, where we do hope to see some hard substrates that will host a completely different set of fauna. Ones that need that hard substrate to attach to, and that will be crucial for their survival. Looks like we're going into an area where the mounds are increasing. I have high hopes we'll see a Bathynomus. Ooh, yeah. Ooh, can we have a quick zoom in this burrow? There's it looks like there's an animal in there as well as an Ophioid. Please.
Go ahead in.
Okay, maybe maybe the burrow's collapsed, but there definitely is a brittle star or Ophioid here within this burrow. Perhaps this is an area where it's a little more sheltered. The pilot did say there is some current in this area, and so perhaps this is a little nook that it can call its home. So with that, I'm going to nip off to lunch and leave you in the very capable hands of Chuck.
Enjoy.
Come on in. No.
We lost our clock. Yeah.
We lost our clock.
Oh.
It's back.
And we're back, and I believe this is the Dicrolini that we saw earlier with those wonderful elongated pectoral fin
Okay,
rays.
come on up.
There was a midwater fish zoomed by, might have been a viper fish. There's a large fish, relatively speaking,
Go ahead in.
above the bottom. And that's another macrurid. Nope, I can't quite tell from this angle. Well, it's got
Get rid of the lasers.
interesting
Sure.
elongated fin rays on its pelvic fins. I'm not sure if we've. Is this? Well, we'll have to see if anybody comes up with anything. That's got huge eyes. I'm not sure we've seen this
We're going to
before.
get a
Is
loop.
this
No, I think we're okay.
I don't think it's
Never mind. Go up a little.
No, it's not. I don't think it's a rat tail. Anybody on shore can help us, that would be great.
Just doesn't want to cooperate.
Of course, it's not giving us its best side for identification.
Yeah, come on out.
Okay, and we're looking down on you.
That
Okay, I'm going to
the
do
fact
a
that it
turn
just
to my
Okay,
ran
sure.
starboard
into
here.
the bottom
Copy that.
suggests we may have blinded it. Fair number of fishes here.
Just cleaning myself off here,
Once we
Chuck.
get out of our own cloud.
See if that helps.
Quite a number of larger mounds and their adjacent craters here, which suggests that something is excavating below so that the sediment collapses and forms a crater, and then the animal below
Copy that. Thank you.
defecates out the sediment that it eats, the unwanted mineral portion and waste material, which forms a pile. Adjacent, there are
Let's come in
worms
right
in
here.
shallow water that do that, the lugworms, for example. And that looks like a bit of sargassum.
Yep, come on in.
Yes.
Come on out.
Sargassum, of course, is the brown alga that floats at the sea surface, and eventually, not sure what the circumstances that lead to its demise are, but clumps of it will eventually sink to the sea floor, where they provide nutrition for a
Get the
variety
fish.
of organisms. And here's another eel. And again, I'm on thin ice with this one. I think it's a synaphobranchid. And we are having an ROV crew change here, so hang tight, folks. We'll be on our way shortly.
Some very soft sediment.
Yeah.
I'm going to pick up a little bit here.
Okay, copy that.
The crew, current crew
Sinking
is
again.
discussing details with the folks who will replace them.
Just getting set up here. Okay. 230. All right, watch lead. We just had a shift change.
Getting a little
Right.
high.
We still have one person that we're waiting for, but I think we're ready to continue. I'm going to get us out of this dust cloud. I'm going to push out a little bit, Jeff.
Copy that. I'm lined up on 230, Delta 11. All is looking well.
Okay, coming down. Pushing forward just a touch.
Ship's currently moving, or we're still swinging?
I think it's finished. That's what he said, but we can confirm.
We can get more going once Fernando sits down. I don't have a general uphill. I don't my sonar, so I guess we're just heading for waypoint 1 new.
It's very flat.
Roger.
This whole area, but yeah, I think waypoint 2 will be off to the west.
Okay.
And then we'll hit the ridge and then work down south along the ridge. I think is the plan.
Good.
Hi, Fernando.
Hello, Dan.
So, shoreside, we just had a shift change in the front row. This is Dan Rogers, sitting pilot.
My name is Fernando Aragon. I'm sitting as navigator.
Sitting co-pilot is Jeffrey Lanning.
Over on the right on video, Bob Knott.
And in the back is Caitlin Bailey.
And we're all engineers and videographers with the Global Foundation for Ocean Exploration.
And just as a reminder, I'm Chuck Messing. I'm the co-science
Okay, Fernando, I think we're ready for a move.
All right, we've been moving 230.
230, yeah. Let's go and do 30 meters. Or
fellow at the
or more.
50. Let's do 50.
All right.
And to my
50
left,
meters coming up.
Hi, I'm Lauren Jackson with NOAA's NCEI, the National Centers for Environmental Information, and I am the sample data manager here today.
and without her, we'd be totally lost. It's true. She takes care of making the labels, among many
Sorry,
other
Bob,
things. She
I'm having a
takes
hard
care of
time hearing
making the
you.
labels,
I'm just going to get
specimens
adjusted
we
here.
collect, and keeping track of all of the data.
Okay, can you repeat, Bob?
That we record, the names of the different
Okay.
species that we observe, and so on. And so we will be heading
Okay.
a little bit
We can,
south
zoom
and
past the
west
skids
for
if you want.
a while till we reach one of our waypoints. This is a rather flat terrain. There's another Medusa
Oh, I see it.
pumping itself against the bottom. And Another Paraphila. Paraphila
Sure.
is a true medusa in the group Scyphozoa, and it belongs within
Can
that
do a partial zoom.
major group to the
Give you a little leash here.
Thanks.
coronates. The coronate
Go ahead.
medusae, because you'll see as we zoom
Dust
in,
cloud is looming,
of course,
just
it's
so you
upside
know.
down,
Okay.
pumping feverishly against the bottom, and it has a groove around its conical umbrella, which is characteristic for the group. Not sure why it's doing this. Marsh Youngblood has mentioned to us that some of these may hang out, or at least the benthocodones may hang out in the flocculent surface material.
Seems like a lot of
My
energy.
guess is that
Yeah.
this
Can zoom in a
medusa,
bit.
and this is just a wild guess, that this medusa
Hold there.
is trying to migrate into deeper water, because unless it's somehow injured that we can't
So
tell,
well here.
because they do migrate vertically up nearer the surface at dusk frequently and down into deeper water
Yeah, it's come
at
out.
dawn. Thank you, pilot. And maybe somebody can fill me in on the details. There's a nice burrow in front. And there's more of that
You're out on
anoxic black
out on
sediment.
the end of your leash there.
in the
So
Coming back a little bit.
and the
starboard
white
or something.
bacterial
Instead of
mats
head straight ahead, there's a cloud there.
indicative of subsurface
Yeah, I'm just going on the other side of it here.
seep activity.
Look at this.
Some squid ink.
Yeah. Looks pretty new.
Stirred up a little bit of sediment during the swap. We'll get out of this.
Give you a spidge more.
It's a little better. Do we know what these white patches are?
Yes, the white patches are bacterial mats that take advantage of the anoxic black sediment and the chemosynthetic activity, or the seepage from below.
So it's been well documented, this dive already?
Yes.
Okay. What's that out in the distance?
That's a good question. Let's take a look at
We'll
it.
just
see
do a
what
quick
that
zoom.
is.
Oh, yeah.
That is another bit of freshwater plant detritus. It
Okay, we
looks
can come
like
out.
water
Thanks.
hyacinth, which is an invasive species that is a bit of a problem
Got a little
in
leash.
many parts of the American South, and that we've been told washes out the Mississippi River, I'm sure among other rivers.
See that? Can do
That
a
looks like
partial zoom.
another. Yep. That's another Paraphila. Again, against the substrate. If you've never seen a plowing jellyfish before, you're seeing it now. I'm certainly unclear about why they're doing this, whether they're just trying to migrate deeper into the water and come up in an area where it's shallower than they want to be. If anybody in the chat room or via email can let us know if there's a reason for that. It seems like it's unlikely that it's injured, because we've seen at least 3 do this on this dive.
Yeah, that's
Thank you,
interesting.
pilot.
Okay, we can come out. pushing ahead a little bit.
I have a guess on that.
Do you?
Our depth is now 796 meters.
Maybe. It's true.
And there looks like the seafloor may rise up a little bit in front of us.
I can't. Oh, you're not talking on any
there you
channels.
are.
There we go. Yeah.
There's still about 10 meters left on that move.
Fish up.
Your left.
Yeah, we should
There's a,
probably keep it moving.
Copy.
An interesting fish hanging above the bottom.
Where's waypoint 2 at?
It's westward.
Can do a partial zoom. Oh, so are we
That's pretty much
about
270.
They were doing
Another
230.
one of
We should check in with
these
watch lead and
eels.
see
I'm
what their
guessing,
plan was.
Synaphobranchid, but I'm hoping
can't
Ken
get to waypoint
Sulak
1.
or
That's what they were going
Mark
for.
Benfield
And then just start
may,
moving directly west from here.
Yeah, I
or
guess so.
someone else
Yeah.
may
Could start
log in
going.
and tell us what
You can
that
zoom
is.
partial,
And
or
there is
more
another
partial.
parasite
That's fine right there.
attached to its flank. That's a parasitic copepod. Copepods are crustaceans, and the majority of them, of course, are important planktonic species. But the group also has numerous parasites of fishes and of crustaceans. At least that's what it appears to be. Well, I'm not sure now because it looks completely flat. There doesn't appear to be any thickness to that white smear
Let's come out a
across
bit.
the side, so I may have been wrong about it being a parasite.
Just a scar.
I'm going to zoom in a bit and try to get the white patch. Hold there.
Maybe that's a scar rather than a parasite. That's excellent. Thank you. Mark Benfield suggests that it's Synaphobranchus. And you can really see the lateral line along the side there.
Okay, let's come out.
And it looks a little bit on the well-fed side.
So we're just going to head west toward waypoint 2.
Hold on 1 second.
Okay. You want to snap zoom on this fish? Should we just get lined up on 270? Okay.
Go ahead, Rich.
That looked like another dicrolini.
Yeah, go ahead.
That black fish along the bottom.
Just make sure the center of rotation is at 8 frame. Thank you. Yeah, so right now waypoint 2 selected is showing 268 as the bearing from sled.
Okay, so we'll get lined up on 270 or so.
Okay, and then once
Turn my heading as you go across. Or skate.
right now the ship is doing a heading change. When they're done, I'll call in maybe another 50-meter move.
Sounds good.
So we have a skate here. If
Go ahead and
we
do a
can
partial
zoom in
zoom.
on that before we take off. I'm not sure if we've seen this kind before.
Hold there.
With those two bright white dots on either side of its eye. If anybody ashore can help us out with this spotted customer. So this is a skate rather than a ray.
Let's come out
If we
just a
look at
touch
its
video.
tail, we can see that there
Right there.
are a couple of small fins on its tail. I'm pretty sure I saw them. And yes, there they are. So rays, true rays, do not have those. Skates do. And this appears to be a female. Can we have the lasers just take a
Sure. Lasers on.
quick look, please? Tell how large this is. So those lasers are 10 centimeters apart. So this ray looks to be close to, if not
65 degrees.
just over a
Looking good.
foot across and perhaps a foot and a half long.
I think the ship's stopped at this point.
That is correct.
Really interesting looking fish. Those white dots are really quite striking on its wings, on its pectoral fins. Thank you.
Let's get lasers off.
We'll see if we get a call in about what kind of skate it is.
Hold there.
It'll almost
Just get a few
certainly
seconds
be
without the
something
lasers.
Raja. R-A-J-A is the suffix for a large number, for a fair number at least, of the
Okay, you can
rays.
come out.
Alright, you're pretty much under me, so I'll just turn towards the west
Okay.
here.
I'm facing 270 now.
Set point at 270, heading still coming around.
Okay.
And there it is, 270.
Looks like a fair amount of burrowing activity
minus
here.
5 degrees.
Probably sizable animals buried in the sediment. You
Okay,
could make
and
these
probably
big
calling that move whenever you're ready, Nav.
We already
mounds and
called it in.
Called it in,
So
okay.
yeah,
Great.
they're moving
Can we
another
zoom on
50
that?
meters, 270.
Fish to the left,
Sure.
please.
Let's do a partial zoom. We'll keep moving in toward it.
We've
Hold
seen this
there.
already, haven't we?
I believe so. Is this
Yeah.
the Iliophis? Or
I
was
am-
that something else? No.
To be honest, Ken gave a whole bunch of IDs.
Right,
that
and I can't remember. I don't think it's the Iliophis.
So that shaking behavior, which we just saw, I've seen fish do in the abyssal Pacific, and we hypothesized, we don't know for sure, but we hypothesized it was in response to the bright lights being shone at it, or it could be it trying to relieve itself from parasites in the gill chamber.
This is the second fish we've seen in just a few minutes with scarring on it. We saw a synaphobranchid eel with a white scar mark on it. This one looks like it's been through the ringer.
Okay, let's come out.
Maybe it's a bad neighborhood. There's a large burrow there. You can see the sediment, the gray sediment dug up from below the surface, having been excavated out and pouring down this shallow slope here.
Can we check out that crab in the distance, please?
Sure. Any interest in the other fish?
Sure, if we're going to zoom there, we could just zoom past it.
Okay, I'm actually going to drive forward a bit.
Looks
We can
like
do a partial zoom.
Midwater fish. of
Yes,
some
it
kind.
does.
And hold there.
Big eyes. Look at those eyes. If it turns aside, we might be able to tell what it is.
Holy cow.
Although I'm not sure.
Those are very disproportionate.
No.
Yeah.
To me, it looks like a deep water cardinal
That's what I was
fish
gonna
with
say.
Okay.
the opposing dorsal and anal
Okay.
fins and epigonid.
Exactly. So those would be in the family Epigonidae.
Thanks, Lauren.
Just doing some evasive maneuvers there. Okay, we can come out.
And
The other one looks the
second
same.
there.
Oh,
Or is
just
that
off
different?
to the right, there's a bunch of bacterial mats.
Okay, let's do a quick zoom. We'll confirm it's the same one.
That's a similar one.
Similar.
We are in 795 meters.
Let's come out a bit, video.
A characteristic of this family is the elongated caudal peduncle.
What does that mean?
The
Hold there.
area right before its caudal fin, its tail, they don't have fins.
Okay, let's come out.
Ah, yeah.
Yeah, there's quite a distance between the pair, the dorsal and anal fin, one above the other, and the tail fin. Oh, well, look at that. With Diva's chemosynthetic experience, I think she can smell these from a distance.
So for the past couple of dives, we've been seeing many of the chemosynthetic habitats in these shallow depressions. Sometimes some are much bigger. And they're very easy to spot from some way away because you can that white bacterial mat actually glows almost in the distance. So here we can see there is some chemosynthetic activity still going on. We can see the reduced sediment in the white bacterial mats, but we're only seeing dead clamshells.
Hold there.
Not much of the other animals. You know, you get mussels where there's a very vigorous flow. There are a couple of these tiny gastropods. I'm not sure if they're tour gastropods, but not much other than that. We're not seeing tube worms. We're not seeing mussels.
Going a little tighter.
We're seeing dead vesicomyid clamshells, and so that could indicate that this area was once more active than currently.
And there's a small gastropod just off right of center, and if you pan a little left, we have a larger gastropod. It's still quite small.
Panning left.
You have to pan back a little bit. There it is. And
Going on that video.
if we zoom in, you can see its little siphon. That is a elongated flap of tissue that folds around, creates a tube that allows these snails to burrow and still act as sort of a snorkel above the sediment surface, so that they can both take in fresh seawater
come out
for
video.
breathing,
I'm going to push
and
ahead.
also for sensing the chemical environment around them.
Looking at you, 85.
85, pushing out.
Daisy chain's a little loose on the BSR here, huh?
Small signs here and there that underneath a lot of the sediment there is probably a lot of hydrocarbon-rich fluid moving around, as well as leaking in some areas, but not the kinds of communities we've seen on past dives during this expedition, as well as that are known from further north of here. Just north of here, the largest brine pool and some of the largest mussel beds and tube worm colonies in the entire Gulf of Mexico.
So do we know what creates these burrows?
We haven't actually seen anything yet. We do know these are biologically made, but we haven't actually observed any of the arthropods, we're going to assume, living within them. This one that we're approaching looks like the biggest one we've seen yet,
Zoom
though.
past the skid.
Indeed.
That's good. Thanks, video.
There's 45 degrees on the camera.
Okay, thanks.
That's quite a crater.
We're pretty much
Oh,
done
but
with
there
that
it
move.
is. Something's at home. Oh no, that's a crinoid.
Is it? Or
That did not make that.
No, it did
hole.
not. Or a stalk of some sort.
Can do a zoom.
Are those tube worms?
Let's hold there.
Oh, that's messed up. So because we have multiple lights, they're shining on the two tubes, which make it look like it's a
Oh, yeah.
multi-armed
Yep.
crinoid, but in fact it is just two tubes of some kind of
Thank you,
sublinid.
Bridge.
You can see what looks like the red
Co-pilot, we're holding
worm
station.
body within a
Copy that.
transparent tube. The red color being
Pilot
powered
looking at
by
you, 75.
Okay.
hemoglobin, which is used to transport oxygen and other chemicals around the body.
And there is a little brittle star tucked in at the base.
Okay, let's come out.
So we've recently seen, yesterday certainly, and this one today, multiple, at least two tube worms apparently growing out of the same base. That looked like a
Is that a new fish?
midwater fish.
Swimming away
Not
pretty
sure
fast.
if that was a viper fish or not.
It did look like it. Is it just me, or does it seem like there's a lot of particulate in the water column?
It seems that way to me as well. And I haven't touched down in a while, so I don't think I'm stirring that up.
Bob Carney suggested that that snail we saw mostly buried with its siphon sticking out might be in the family Buccinidae, which also includes cold water whelks in particular from shallower water.
Okay, I think we can do another move when you're ready now.
All right, another 15. So
Sure. Sure.
Whelk is another term like jellyfish. It applies to a lot of different things.
Is it a squid?
No, it's something else. I can't tell what that is. It's a fish with barnacles on it or something.
Oh, another more squid ink.
Yeah.
Yep. Squid ink in the water. Interesting how it just holds together in certain respects. It's also difficult to tell whether these mound and crater pairs are currently occupied, or are they left over and their resident either died or moved on and built another one.
You want another one?
Because these
Due west?
features
Yeah.
of the seafloor
Yeah, I
may
just called it in.
last,
Just called it in, yeah.
no,
Yeah.
I don't know enough about it. They may last a considerable time after the longevity of their
Gonna
builders.
scan side to side here. So have you guys seen many corals today?
We have seen 0 corals.
Zero
today.
corals. Okay.
Well, we're about to change that.
Because the Jeff Lanning guarantee. Hopefully.
Another one of these ophidiids down the bottom. We've seen a couple of these for this expedition and this dive so far. Oh, this is an interesting burrow here.
Yeah, so you see of those 3 mounds right there, right, the one above and the one below are partially covered with this organic tan flock, and the one in the middle appears to be the active one, with new sediment having been dredged up from below. And little trails.
It's that yellow patch there. Let's do a quick zoom.
What bacteria do you think, Diva?
Yeah, maybe just some kind of different bacteria we haven't seen yet.
You can come out. The Z-bias is a little light.
Vehicle must be a little light because that looks like a lot compared to usual.
Got 24 down.
No.
This is interesting here. You see the bottom in front is almost completely smooth with scattered trails, and then you have a couple of large burrows, and then all of that small activity there, those little piles or pock marks. I'm not sure what they are.
If you don't see much, let me know if you want to speed it up to 0.3.
So the populations can change very quickly. I should say the communities of infauna can change quite quickly.
I think if we just keep 0.2 not moves going, try not to stop in between.
All right, sounds good.
And then if we do go a while without zooms, maybe we could speed it up. I'll just kind of scan back and forth.
A couple of little bacterial mats.
Oh, yeah.
Again, looks like a little, very small midwater fish.
Oh, something in my manip cam.
Oh yeah, you've got a.
It's come full wide.
Medusas.
I don't know if it's real close. Oh.
He's
So folks at
latching
home,
on.
you won't have a view of this, but there's actually a
Trying to shake
jelly
him off.
stuck
Yeah.
next to
Oh,
one of
well.
the ROV's manipulators. The starboard manipulator. It's coming with us for the rest of the day.
Yep, might have a sample there.
Here's another example of that. This is, I have no idea what's causing this. You'll have smooth sediment and then all of these little small bioturbation structures, little, I don't know if they're craters or scrape marks or tiny mounds, and we're out of them now into those low mounds. Most of these look unoccupied. They're covered with the organic flock, but there's an active burrow right in front of us with a tunnel. We have not seen the occupants of those in our last several
Something off in the
dives.
distance there.
And there was something to the right
Yeah.
swimming as well.
We'll come back to that.
Oh, another one of these paraphila.
You want to do a quick zoom? Yeah, hold there.
That's exactly right.
At least he's not stuck
So
on the
Mark
sediment.
Benfield has suggested that another water mass may have been that these jellies are likely from another water mass, and they got carried into shallower water, and now they're trying to get a little bit deeper again. But
Not having much success.
I'm not convinced of that. Poor little guys.
Well, it's certainly unusual. What might have driven these, I don't know what their normal bathymetric range is. I understand they do migrate. I believe that's true.
Yep.
And my earlier suggestion, I can't think of anything else, is that they were somehow trying to migrate into deeper water and ran into the bottom instead.
Do you not think they have more cognitive abilities to realize they're not actually going deeper and they just plow it into the seafloor?
You would think, but I don't know. That's what makes this so curious, because we've seen it over and over and over. There's at least half a dozen instances of this with this medusa.
Okay, let's come out.
This species, Paraphila.
Well,
and we
we'll let
also
him be.
saw a siphonophore earlier in the dive, which was on the seafloor as well.
And we are heading west. Up a slope, a slight gentle slope.
So do the bacterial mats suggest the presence of hydrocarbons, or?
Exactly. So
Okay.
the black reduced sediment suggests some kind of organic enrichment that was then utilized by bacteria, and in the process of doing that, they depleted all of the oxygen in this area of the sediment, and that resulted in a state change of the iron.
Copy, bridge.
And the iron, the state change has resulted in this black iron compound being present in that sediment. And then a different type of bacterial mats moves in to that environment, which is the white bacterial mats we've seen. And so that's why they're only found in reducing areas.
That's how it goes. That's an interesting story. That succession.
And that takes place at all chemosynthetic habitats: whale falls, wood falls,
Wow.
cold seeps, and hydrothermal vents.
So the white ones are not pioneers by any means.
Well, they are in terms of the grand scheme of
You wanna
things.
do a
They will
partial
be
zoom?
one of the first settlers, and they are what most of the animals
hold
will
there.
eat at these habitats. But
Now, I am not sure.
doing
what
some
this-
yoga on a Sunday morning.
Well, it's a spider crab, but that doesn't tell us.
You can do a little
great
tighter
deal.
zoom.
I don't think it's Rokinia.
Hold there.
No. This has got, well, it might be related. It's got a small forked rostrum.
Tilting down, just leaning off the right side there a little bit.
And as it moves its legs, you can see how cohesive the sediment is
77%
clinging
down.
to its legs.
Or 77 degrees, sorry.
I don't know if anybody ashore can help. I don't think this is Rokinia crassa.
Can we have the lasers on, please?
Sure.
Corbett?
Let's come out for lasers video. There you go.
Yeah, it's not that.
I mean, that's pretty big if you stretch
Yeah.
out those legs.
Well, that's true. Probably.
That's 40 centimeters across if you stretch out the legs,
yeah,
leg
that's
to
true.
leg,
That's true.
at least. So this will be one of the main predators in the area. You can see it's got a very large pair of claws.
You want to go in
And,
tight again video? And hold there. That is an ugly crab.
Wow, beauty is in the eyes of the beholder.
He's well
He's just a little
camouflaged.
dirty, that's all.
Yeah.
You could brush up pretty nicely, I'm sure. When he
Gotcha
molts
at
away,
almost 90.
Okay.
sheds that exterior skin, probably looks like $1 million.
Yeah, he's just close to molting, Dan. Jeez.
Okay. I won't be so quick to judge
Judgmental?
crabs. Yeah.
This is one of the first times we may have seen one this big, perhaps?
Well, it's
Come out
different
just a touch
species,
video.
I'm almost certain, because look at it's got these
Hold there.
these stout spines at intervals all over its body. I'm not. It just doesn't look quite like the Rokinia that I'm familiar with.
Just a little longer.
Okay. Just have a few more seconds, then we'll have to shoot ahead.
about, well...
Oh
boy.
Bummer.
I've got a photo here of Rokinia crassa.
Nope, he's not
that
bothered.
Looks like it might be this species after all. Certainly Rokinia crassa is local.
You can see how light and flocky the sediment is here, though. How much it's
losing
stirring
you
up.
there.
Okay.
Just
All the way
walking along
back.
the sea
Okay,
floor.
just a little more.
And how
I'll push-
deep it's
push
sinking
ahead.
into that sediment. Bob Carney suggests this crab may have been in a burrow, and that's why it's so covered in sediment.
Okay, maybe we'll let him go. Let's come out, and then we'll push forward.
A first for this dive, I think. Yeah.
Could crabs like that be creating those burrows, or is that not typical of their behavior?
Chuck and I just looked at each other like, we don't know.
Okay.
I did not hear the question.
Oh,
sorry.
could the crabs be creating those burrows?
Those
or is
big
that
ones?
not typical of their behavior?
Those burrows with the great big mounds and the craters next to them look like something is living in the sediment and bringing up fecal material to make those mounds.
What just kicked up that sediment?
Somebody made a quick getaway.
There's a fish coming into view. Another one of those Snaphobranchus, but there's
You want to see the one on the left? Okay.
Yeah.
Do you want to do a snap zoom
Color
here?
is what's throwing me off with that one.
Snap zoom, and then we'll go to the other one. Hold there.
That looks like the same as we've seen.
Yeah.
Although this one's got a little tail to it. Is that a different... and this is the one with the long nostrils. Is it got the long nostrils? Yes. Is that the Iliophis?
I am not sure.
Let me
Again,
see if I can check.
Ken gave us a whole bunch of
Okay,
different
let's come out
names.
and get the other one.
I'm not sure if
You're
that
looking
one's a
good on
congrid,
time.
then it would be
Great.
suggested Bathyuroconga, or that could be Snaphobranchus affinis.
The other one gone. No, he's still up there. I think that.
Oh, there's two. Let's get a zoom on the one in the water column heading
okay,
towards
in the middle.
bottom. Yeah,
Okay, go ahead.
please.
Hold there.
Is that a baby halosaur?
Oh.
Again, very out of proportion. This really long tail exhibiting that classic halosaur movement, but the rest of it doesn't look very developed.
I don't think that's a halosaur. Halosaurs would have the that duck bill, wouldn't they?
That's true.
Okay, let's let him
Maybe a
go.
little
He's getting under
macrurid.
me.
But that tail movement and the proportion of the tail compared to the rest of the body, very interesting.
We'll get
Doesn't
this
look
guy
a dorsal
here.
fin to be a macrurid.
That's true as well.
Looking at
So
you,
basically
70
we don't know
degrees.
what that is.
Might
So
be a
I
Let's do a
custody
zoom
hawk.
on this fish
Yep. I think that last eel on the bottom may have been Iliophis.
and hold there.
It's got those nostrils at the very tip, and it does have a little fishy caudal tail, caudal fin. And I wonder if this is another Dicrolini. It's different than the one we saw earlier, but it's got those.
This definitely looks like a macrurid. I'm not sure. I can't say much more than that, but
Can do a
has
tight
the
zoom real quick, and then
very,
we'll have to go.
Dorsal, the characteristic dorsal fin,
right.
as well as the very pointy face.
But this one has got an not only the elongated pectoral ray at the very
Okay, let's come out.
anterior, or the first pectoral ray, but it's also got a couple of elongated
Yeah, we'll come full wide.
pelvic fin rays.
And
Just
the
on the bottom
coloration
of the
is very
screen. Yep.
different
That's
to
71.
anything we've
Push
seen
ahead.
before, I think. A high fish diversity on this dive.
About 5 meters left on that move.
Okay.
I suspect that if we keep moving upslope and seeing these, what is that?
And do a quick zoom.
That's a nice siphonophore.
And hold there.
Look at that, with that long row of nectophores, swimming bells at the anterior end. There's a tiny little gas gland, and then the chain of zooids for feeding and reproduction. So this is a physonect siphonophore with that combination of features.
Maybe Irena, the genus Irena.
Yeah, I don't know them.
So ways out there if you want to push out.
I don't
out
think
of the
that
dust.
fish we just saw was Dicolini.
I think we'll let it go.
Else.
Push ahead and come out
Definitely
video.
something else.
Camera's at 80.
Good copy.
I think we're okay.
Coming down a bit.
There's 45 on the camera.
Okay.
And Mark
looks well.
Youngbloth wrote me that the Paraphila, the conical purple jelly or medusa that we've seen banging into the bottom is a typical behavioral response to bright light.
Do you guys want another
So that
15-meter
explains
move?
that. It's
Sure.
It's extremely negatively.
Oh, what is? Oh, no, that's sediment.
It's extremely negatively phototactic. It doesn't like light, and where it's been studied in the Norwegian fjords, about 85% of them migrate daily throughout the year, moving from 400 meters all the way to the surface. And of course, the surface waters in Norwegian fjords are much colder than they are here. So I can understand that these guys are found in deeper water than they are found in the in places like the Norwegian fjords, where of course we have much warmer water near the surface. And sometimes he says during the summer, when there's a pronounced warmer layer, a distinctive warmer layer near the surface, they'll just come up against that and stop, and they won't go any further towards the surface. That abrupt change in temperature with depth is called a thermocline. And I'm sure many of you have felt the thermocline, even if you've
is that a creature?
not necessarily in
I'm
the
gonna zoom
ocean.
Partial.
Although scuba divers encounter it all the time.
Hold there.
But in freshwater lakes, you dive down in the summer, and you're nice warm in the surface
Don't think so.
layer,
We can
and then
come
all
out.
of a sudden you feel colder water below.
On this object?
You've experienced a thermocline.
Okay. Let's try to go in here. Okay, go ahead, video. Hold there.
Well, that's interesting. Is that a xenophyophore or what? No.
I'm not sure what that is. It could be dead sponge, degrading, covered in sediment. It could be a rock covered in hydroids and other sediment. It could be an animal that's covered itself in... I have no idea.
Nope.
Okay, thanks very much, pilot.
Okay, and come out. Something on the left. Looks like a stock out in the distance too. Potentially. No, never mind.
Nope, that's in the water column.
Just in the water column. You want to zoom in on this here?
Sure, I think it's a stick. Or a tube. Okay, that's interesting because it looks like a tube worm tube, so perhaps a lamellobrachia tube, and that's one of these chemosynthetic tube worms. But then you can see where it's sort of slipped down the hill or been carried down the hill. Definitely has a trail associated with it.
Okay, let's come out a bit.
Quite a couple of fish around here. We've seen ophidiids, macrourids, midwater fish, synaphobranchids. Quite a diversity here. Hypnopnids. What is that? Just going out of screen, there's one of those tumbling snails to the bottom.
Oh, really?
Right.
Gaza.
Yeah,
The bottom
Gaza.
right.
Yep. Very shiny. I did not just imagine that. Nope, nope. Keep going.
Keep going?
Right. Yep, keep going
Okay.
right. Keep going right. There!
Oh, there he is. Okay. Backing up.
Some kind of snail.
Well, if it was tumbling through the water, it was most
Okay,
likely
you want to
Gaza.
zoom in?
It wasn't tumbling, I just saw
Well, let's
a
see.
shiny shell.
Well, it still looks like Gaza. I've actually never seen it before. The shell is upside down, so you get a really good view of the
Oh, you can see his little
animal.
eyes.
I might try to touch
And,
down here.
It looks like it has
Just come out a bit,
filaments
video.
along its foot. That's something with which I'm not familiar. That's really interesting.
I'm going to get a closer look here.
That sounds good. Thank you.
Oh,
Nope,
he's
flipping
flipping back
back
over.
over.
Ah.
Yeah, that's Gaza. The one species I'm familiar with is Gaza superba. I don't know how many there are here in the Western
Okay,
Atlantic.
go ahead, video.
Tina had look at those filaments around its around the margin of its shell. That is very cool.
Go in tighter if you have it.
And
Okay.
of course, you know, if this particular snail
70 degrees.
Okay.
were to undress, we would refer to it as the Gaza Strip. Sorry.
Radio silence with that one.
Okay, I can come out.
So we did see quite a lot of these in the Pacific, and there they were observed doing this weird tumbling behavior, where they would literally throw themselves off of rocks and then tumble down
Okay, let's
slope
come full
while
wide.
wriggling vigorously. I know there was a paper submitted about it as a result of the Okeanos Explorer exploration from the Pacific. I think Mary Wixton was on that, perhaps Chris Kelly and a couple others, but I can't seem to find it.
Is this one of your epigonid
swim
deepwater
away, buddy.
cardinals?
Yeah, it does definitely look like it. Oh, so the videographer, Caitlin Bailey, sitting next to me, has just said that if you go on their website, www.oceanexplorer.noaa.gov, there's a highlight video of one of these Gaza
Push ahead,
snails
Jeff.
tumbling down a slope. And we just passed a red
Cappy.
ctenophore with its feeding tentacles exposed.
Bob Carney mentioned earlier, I don't know if you picked it up, that tube that we saw lying across the bottom, he suggested it might be a tube of the Polychaete hyalonesia, which makes a rather transparent tube.
I don't think he's referring to the one which we said was lamellobrachia. because there was one that was definitely chitinous and chemosynthetic, and I don't think it was that one.
All right.
And even though we are exploring what looks like an area where there isn't much life, there likely is within the sediment, which is probably very rich in organic matter, various types of food. There are probably hundreds, if not thousands, of species living here, many within very small areas. Usually they're most abundant down to about 10 centimeters, but they can get down to well over a foot or 2 feet in the sediment. Depends how loose the sediment is in this area, and therefore about the grain size and lots of other different factors like that. Of course, using an ROV to explore these types of areas isn't necessarily the best tool. If we were to want to quantify,
you guys have seen this already, right?
Have Brachiopods viridensis. So if we were to want to have a look at what were the main inhabitants in this area, we would likely use something like a box core or a megacore. And with that, we'd be able to actually bring up a big chunk of the sediment, and then sort through it with sieves and so on to be able to look at those much smaller animals living here. And of course, many of those are likely unknown. As you go down in size class in the deep ocean, get smaller and smaller animals. Those animals are much less well known than the megafauna, or the animals that we tend to be looking at using our ROV. We're just going over
Fernando,
the brittle star.
before we get another one in, I think we'll do a pilot swap here.
Sounds good.
So if you want to jump in. Yeah.
And if any of those of you out in the ether who have joined us from a couple of our previous days, you'll notice that we have not seen much in the way of sea cucumbers here. Unlike the situation where we were a few days ago, I believe most of those purple sea cucumbers were in deeper water.
It's looking a little, yeah, there we go. Looks good. All right. You want to go ahead and call in a move, pilot?
Yeah, I think we're ready.
All right. Bridge, this is RV Nav. Yeah, we'd like to do another move, please, of 50 meters. That's 50 meters, bearing 270 at 0.2 knots. That's a good copy.
Marsh Youngblood has let us know that siphonophore we saw swimming by a few minutes ago with the long series of swimming bells or nectophores could be Arena, as Diva suggested, or possibly another genus, Bargmania. And there's a nice shrimp, perhaps in the Metacarcinus.
Thanks, Bridge. And Bridge, this is RV Nav.
We take a look at that shell, please.
Can we please change our bailout direction to due east?
So this looks like a shell belonging to perhaps bivalve from the family
Partial
from the
in video.
genus Atarax.
Hold right there.
And this is a different type of chemosynthetic bivalve. We've not seen one of these yet alive for this expedition, though we did see one of these shells.
Gonna bring the camera down a little bit.
Earlier on. And so this is from the family Solimidae. And they are essentially clams that live within the sediment, again, in areas where, which are very reduced in nature, and they function using sulfur oxidizing bacteria within their body, so endosymbiotic bacteria within their gills, and with those, they are then able to use the sulfide-rich fluids seeping out of these areas and get their food via chemosynthesis, which is where chemical energy is used to create organic matter rather than via photosynthesis, we know, and what we rely on, where plants use sunlight to create organic
Do you want to come in a little bit more video?
And so these likely live buried down within the
Hold there.
sediment. We may be driving over loads of them and don't know they're actually there, but they do have this very characteristic shell.
All right, let's come out.
Thank you, pilot. So it's interesting that shell may have been eaten by something and then they just left the shell there, or it may have died and made its way to the surface. Who knows? Ooh, see, there was one of the gases that's just tumbling around. Ooh, tumbled quickly. It was definitely
you
just
might want to
there,
give yourself
both
a
Fernando
little
and I
bit
saw
more
it.
altitude. You're just dragging around a little bit, causing a bit of a dust cloud, which will be inconvenient when you go to zoom next.
All right.
There you go. That's
So it
probably
looks
better.
like we're finally getting into a little bit steeper terrain. Moving up onto that, or towards that ridge feature where we hope to encounter some corals. A lot of these little patches again of bacterial mats, also some burrows with mounds next to them. What is this little animal we're coming up to here? Just left of the lasers, the pinkish. Ooh, first let's see the thing in the water column. Sorry.
Partial in video.
Right
there, hold.
Comb jelly, I would guess, but not certain. I don't see
Come out a little bit.
It doesn't look like a burrowid.
Where did it go?
It may have become chum in our thrusters.
I think it got blown away.
That's okay. So in front there was pinkish. Yep,
right,
there you go.
Right above bottom.
Is that a Dumbo octopus? Please, 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 please.
I'd be surprised. It's going to be a very, very small Dumbo octopus if that's what it is.
All right, coming partially video. Hold right there.
No, dandelion siphonophore.
This is my first dandelion siphonophore.
We saw one earlier in this expedition.
Come in a little bit more.
Seeing that, I may have been at lunch or out to lunch.
So this
Right
is
there.
in the family Rhodalidae. These are siphonophores which attach via tentacles to the sea floor. They are known particularly from chemosynthetic habitats. They were first, or one of the main places they were
Okay.
first observed
You can
was
zoom in a little
at
bit
the
more if you
hydrothermal
want.
vents in the Galapagos Spring Center in East Pacific Rise.
Bringing out the camera.
You know, you can see the elongated, I guess those are dactylozoids. The normally would function as stinging apparatus, and it's using them to walk along the sea floor. It reminds me of a Macy's Thanksgiving Day parade balloon.
Nice shot, Fernando. Looks good.
Look at that.
Thank
That's
you.
marvelous.
65 degrees on the camera.
All right, copy. Are you okay, video? Do you want any closer shots? Okay, let's come
It
out
looks
then.
like it has a tiny little amphipod or something on its right side.
All right, sounds good. Lifting off. Bringing camera up.
Beautiful. Always a treat to see those.
Just off bottom right. You're looking good. A little push out that direction. You'll be right in front again.
I think the most abundant animals we've seen during this
Copy.
dive have been fish, and then after that have actually been some kind of cnidarian in the midwater.
Yep. There's Bathypterois viridensis again. Absolutely a handsome fish. A very bizarre, alien fish, but with handsome markings.
I think they got a couple of these so we can give a push, keep up with the ship.
All right.
Well, actually, this is Nav.
Go ahead, Nav.
I just wanted to let you know that at this pace, we're going to reach waypoint 2 in approximately 1 hour. And then we'll have 2 additional hours to make our way along the ridge toward waypoint 3.
Bringing camera up.
Okay.
All right, I'm pushing out.
Thanks, Bridge. Stand by.
There's 45 degrees there on my camera.
Copy. 45 degrees.
You ready for another one, pilot? Do you want to wait a bit to catch up?
I think I'm.
So many fish in this area.
I think he's 45. I think we're good. Pushing out as well.
50 meters, 270.
So you know you're pushing towards the south. We're heading towards the
Oops.
west.
And as far as we can see, just sediment with the odd
Interesting
fish.
feature there.
But this is
All
an
right.
interesting ridge here. Look at this, so straight. That's neat. Just drops away like that. Doesn't the ridge doesn't appear to be a dune built up by seafloor current. Maybe that the depression is associated with some change in the subsurface
And it looks
architecture.
like a Chasian crab we're just maybe about to pass. We've seen a couple of these during the... maybe it's not.
Partial in
We've seen
video.
a couple of these during the expedition, if it is, Chasian feneri, but this one looks a little stockier, perhaps.
Video partial in. There you go.
Oh no, so that is not Chasian feneri. That looks different to the one we were looking at earlier. This one has some very thick arms.
Oh yeah, look at that. This is a spider crab in the broad sense of the word. Again, that family has been divided into 6 different, that former family, Magidae, has been divided into 6 different families. This looks sort of like a member of the family Mithracidae, but I don't know if it has deep water representatives. I don't know if that could be one of these or not.
All right.
That's great.
Let's
Thank
come
you.
out video.
All right, I'll get that move in.
Big claw.
Okay, sounds good.
Bridge, this is ROV nav.
Bringing camera up.
Another move, please,
Oh,
of 50 meters. That's 50 meters, bearing
Look at how
270
regular
and 0.2 knots.
That break in the sediment in the seafloor is.
Any interest right here?
That's a good copy.
Watch lead.
Sure, we can get a quick zoom. This looks like
Let's
another
come in
halo
partially.
Yeah, this one does.
Right there. That's good.
And just past it is.
Somebody's
Can we
lying
get
on
a
the surface.
zoom of that because
Okay, let's go for the other one.
sedentary nature of that fish is
Oh.
a little strange. Haven't seen one of those before. So that to me looks like an eel pout.
Let's come out
Yes.
video. I'll
which
get
is
closer.
in the
Thanks,
family
Bridge.
Zoacidae, and those tend to be. No, that's a lie. I was going to say tend to be found in chemosynthetic habitats. But
Video,
that's not
let's
true.
come out.
They are one of the main inhabitants
Thank you.
of many chemosynthetic habitats, but they are known else from other. They are just normal scavengers at various depths in our
So you can
world
see that
deep
ridge
ocean.
feature on the sonar.
But this looks like one that we may not have seen yet.
It's cool.
It's cool.
And we've got little tracks and trails running up and down the and across this this muddy face here, this mini escarpment.
All right, let's come in a little bit.
Okay, coming in.
Zoom in. There you go. Right there. That's good.
Yeah,
Okay, it's
that's
partial.
what,
Beautiful.
you're right on there.
Nice work.
You
So
wanna
these fish
come in a
tend
little
to
bit tighter.
Just lie
Okay,
on
coming
the
in.
seafloor or rest
Right there,
amongst
that's good.
mussels in
There
a
you
mussel
go.
bed.
Yeah.
They're not very
Looks good.
active. Several species I know of from cold seeps actually eat
Doesn't look
many
like it
of the
has
small
a pupil at all.
invertebrates in those habitats.
It says one huge pupil.
All
Not
right.
sure what species this is.
I think I have more zoom.
If you want,
Or
yeah,
more
you can come
genus,
in.
Maybe
Okay, coming in.
Pachytara, but really not sure.
And just as a reminder, as has been the case for most of the bottom-associated fishes we've seen today, they tend to be
All right,
elongate,
whenever you're good,
eel-like
video.
or
Okay.
tadpole shape,
Coming out.
and gray, dark gray,
All
or
right.
pale
That's
gray.
forward.
And this is
Thank you.
characteristic
Lifting off.
of many of the deep water bottom-associated fishes around the world. Not absolutely every one, of course. And this stands in strong distinction to the midwater fishes, the ones that we've seen a couple of them near the bottom. They're typically vertical migrators, and they tend to be black or silvery because at least part of their daily commute or life cycle, they're up in water that is at least dimly illuminated with blue light. The red light doesn't penetrate. And so being black will allow them to blend in with the background.
Are we
and
moving
being
right now?
silvery,
We are moving.
Or being
Okay.
silvery will allow them to reflect the ambient light and also act as camouflage. So the this little fish that we're zooming in now, the epigonid deepwater cardinal fish, is of course breaking a bit of the rule, but I'm not sure how deep they get. Do you know, Diva? Let me look that up. Boy, that's a really straight little sediment escarpment there.
Can we have a quick look at that jelly? Oh, never mind. So there's another chimera in the distance coming towards us,
Boy,
or
you've got
moving
good
away from
eyes.
us.
You've got very good eyes.
they've got this very characteristic movement of their fins.
Slowly flying through the water.
Hearing breathing sounds from somewhere. Okay, yeah, I think it was pilots. It's better.
Is that better? Do you hear anymore?
Bob Carney suggests that
A little
this,
bit, maybe.
This
Your mic a little bit
wall
lower.
we're running along here might be a bottom.
All right.
Bottom
How's
fault
that?
associated
I don't know. I can't hear anything. I never hear
Some
anything.
of
But
That sounds good.
faulting
yesterday,
associated
Roland was
with,
saying that it was probably me.
the subsurface geological structure. This is again looks like
Can we get a shot of the Chimera?
Hydrolagus
Yeah.
alberti,
So let's come in partially.
a small one.
Okay, coming in partial.
Okay. Hold right there. Bringing camera down a little bit. Oh no.
Should I come out, pilot?
Yeah, please.
If you feel like
Okay,
if you feel like
coming
you're
in.
sinking faster than you want to, you can try adjusting the Z bias a bit. You feel like you're floating down into the sediment.
Okay. All right, come in a little bit more.
Okay, coming
This
in.
is definitely a different
There you go,
individual
right there.
to the one we
There
were
we
looking at
go.
before. And
That
this
is so
may be one of my new favorite fish.
absolutely.
So
See,
adorable.
fish are so cool.
And this deserves to be a plush toy.
I'm going to come out just a little bit for the tail.
Copy.
Where's the end? There it is.
Bringing
Pitch at
camera
85
up.
degrees, just so you know.
Copy. All right.
Okay.
Let me know when you're good, video.
Okay. He's cute.
Let's
but
come out.
okay.
Gotta go.
That's full wide.
Thank you.
Let's make a boogie for it. Headed towards the west.
All right.
So we're going to continue to move along this very strange feature here
Pretty
on
much
the right
straight
that we're
down at you
facing.
here.
Copy.
This
That's some nice
terrace
shots of the fish,
or
though.
something.
Rhonda, that was great.
Yeah.
Thank you.
And we're going up and over it. Could we get a quick zoom on some of these shells, please?
Sure. Which ones are the shells? I see a lot
the
of
bright white.
anything
Yeah,
bright
like
white.
cream, hard-looking white shell.
Okay. Come in video. Hold on
Okay.
1 second.
Yeah.
Can you turn on lasers? Thank you. So that one right there.
So, exactly.
Okay, let's come in video.
Okay, coming in.
Oops, bringing down camera.
Okay, coming in more.
All right, go ahead.
So these look like clam shells again. Dead, unfortunately. We can see some fluffy bacterial mats
Still right on
just
top of you,
below it.
just so you know. Getting a good shot, though.
Thank you, pilot.
Thank you. Come out video.
So
Okay, coming
this
out.
again is an indication that
And
at
then
some
catching up.
point
Okay,
there was
that's full wide.
probably a
Thank
much
you.
more pervasive hydrocarbon fluid flow in the area, and that's what these animals would have subsisted off of. That has likely waned, but
Thanks, Bridge.
and left all of these animals which have died without that energy source. And Carolyn Rupels says that in the chat room that she's been at home plotting our locations in her
No problem. I figured that was
GIS,
coming.
and
So you can take your time now. That's
from
fine.
her estimates, and
So the ship just needs a minute or
shows
two
that
to
we
change
are
their head.
about 3 meters
So we'll be here for a bit.
There's
away
something
from the edge of
like
a
straight ahead.
confirmed
Pilot.
gas anomaly.
It looks like it's like long
That
lakes
was a little while
or
ago, but it means
down
that we
lasers.
are in an area
Okay.
which the Bureau of Ocean
Partial
Energy
zoom?
Management
It's a hole.
has
it's a hole?
identified as having some kind of
It's
gas
a
or
hole.
fluid
Never mind.
seepage.
Anything inside?
No.
Yeah, we haven't really seen
I-
anything in any of the
I thought it was an
No,
urchin
no.
from
It looks fresh,
a distance.
but we haven't
Never
seen
mind.
anybody at home.
All right. Okay. Come past the skids. Video. There you go. Thank you. Oops.
So Carolyn's just saying that the Boehm anomalies that she was just referring to are based on seismic data, and so that would probably indicate a shallow subsurface gas in this area, and whether it breaches the sea floor means as a seep is not indicated in that
See the-
data, but if
You're
it
still a
were
little
to come
tiny
very,
bit
very
low. I
close
think you're
to
still
the
dragging a
sediment
skid.
surface,
Mm.
that would result in these chemosynthetic
I know right here
habitats
on
that
your
were,
snip cams, they look like they're up
we've been
high.
seeing throughout
They're
this
up off the
expedition,
bottom.
but hey,
But because
it may
the
just
slope
be,
is sort of coming off your left
a
side
little
there,
too far below the surface
the outside
to
of your
really have
skid
any real
is touching and grabbing,
effect on
which
the
isn't a big deal
community.
other than
But as
when
Bob
you go to
Connie
stop,
has
all
just
that sand's going
put
to
in the
come
chat
with
room,
you in your
we
wake.
are seeing
You
increases
get the image.
in the number
Alright.
of shells as
So
well as
every once in a while when you
bacterial
glance over here,
mats
see
and
how
the
much
reduced
you're
sediments. So
dusting up,
perhaps we are,
and that'll give you an idea how
on the
close to the
cusp
bottom you
of
are.
some
Alright.
much more active
Right there, I'm not too far from the
area,
bottom,
an area
though.
where the
Nope.
seepage gets
No,
very
especially
close
with a
to that
dive like
sediment
this,
surface.
it's actually kind of nice to see out, see what's out there.
Okay.
Always, you know, you can always brighten it up if they need to.
All right.
And I still have you at 75 degrees, so. No rush since the
Bridge,
ship isn't
RB-nav.
moving. But
Let's come
just
in
wondering
on
if you were
this,
comfortable with your heading at
yellow
180.
spot.
So Mike,
Okay.
our
Right there.
Coming
our
in.
mapping lead on board, did not find any gas
Okay,
plumes
we'll stand
in
by
this
for a minute or two
area.
more
He
before we
did
call in
find
a
them
move.
much further south. That's like a sponge.
I have more
Bit of
zoom
weird
pilot.
place for it to be.
Come in.
Okay, coming in.
Right there. Hold it.
Yeah. I wonder if it's encrusting on a shell.
Is there any chance we could collect this? It's just a very weird sponge.
Sure.
Or is it too small?
Come out, video.
Yeah,
Okay,
maybe.
coming out.
Can we have the lasers on before we make that decision, just to see? It does look very small. Actually, maybe let's not bother. No, that's very small.
Also, Marsh Youngblood.
Have you used the scoop before, Fernando?
Never.
No.
But is there
Said
any
that
interest,
the,
or it's too small?
No, let's
watch
carry
me.
on.
Okay.
Actually, sorry about that. Thank you so much.
All right, let's.
That
It'd be a
benthic
pretty
siphonophore we
easy
saw,
scooping candidate
maybe
if you
something
wanted it
called
much later.
Dromalia
Just so
alexandri.
you know.
Go, Bridge. That's a good copy, Bridge. Thank you. So, watch lead, was the sample too small for your purposes, or were you concerned about the ability to grab it?
Both. And plus, I didn't really reach out to the community. There were just a comedy of errors there.
Okay, we'll move on.
Yeah, let's move on.
All right.
I could go ahead and call that move in if you're ready, Fernando.
Yep.
Okay, 50 meters, 270.
Sounds good.
Bridge, this is
I'll
RV
push up.
Nav. Another move then, please. 50 meters, 50 meters, bearing 270 at 0.2 knots.
So again, more of
Good
these
copy.
bacterial mats as we're moving up slope. Lots of fish around here.
And there's quite a
Coming
an
partially
upslope
video.
in the distance
Right there with
Okay.
for the fish that's suspended.
Okay, that's partial.
Yeah,
Okay.
here we have more of those midwater the midwater cardinal
Deep
fishes?
water
Deep
cardinals.
water, sorry, deep water cardinal.
Yeah, I think we can let this one go.
As well
Thanks,
as
Bridge.
lots of these,
Can you
again,
repeat that, please,
Sycamide clams,
Pilot? Sorry, I didn't hear you.
We can
and
probably
two
let it
other
go.
types
Okay.
of fish above,
coming
like
back
the
out?
Ophidiids.
There you go. Thank you.
Really a high fish diversity in this area.
We can probably zoom in past the skids. Let me just bring it up a little bit so we don't see. There you go.
You got another move going? Ship move?
Yeah, I think he just
Called
called
it in
Do a quick zoom here.
2 minutes ago.
for the okay, come in.
On the
Video.
bacterial mats and all those
Zoom in.
shells.
Coming in,
Hold it right there.
holding.
Okay, getting closer.
So those look like little gastropods amongst dead clam shells, I think.
Yes, I would say.
All right,
Do we have
let's
any more
come in,
zoom?
come in a little bit
Yes.
more.
Okay,
On those
coming in
little
more.
snails.
Right there, that's good.
So the ridge snails look like Canoia miroglypta, which are one of the most common inhabitants of cold seeps in the Gulf of Mexico, as well as in the Caribbean region.
These
Is
guys
that full
have
zoom?
little zoanthids
I
on
have
them,
more.
some of them.
Go ahead.
There's
Okay,
one
coming
far
in.
lower left.
Oh, that's full
There's a
zoom.
Galatheid,
So yeah, that's like the Immunodopsis, squat lobster.
and it's on a one of those buccinid type gastropods
Come out.
covered
Okay,
with mud.
coming out.
I think maybe that gastropod
Right there,
is
that's
a
good.
Phymenorhynchus.
The big one with the squat
Yeah.
lobster.
What's
Okay.
this?
Right there.
The one straight ahead.
There's
Yes.
a lot
Okay,
of little
coming in.
gastropods here.
Right there. There you
So
go.
this is
That's a
an
clam.
old clamshell with a Chitoptera tube coming out of it. And then that
Some
snail,
of those snails are alive. The one up at the very
oh
top.
yeah, most of the small ones are alive.
Oh, okay.
I think all the snails we've seen in this area have been alive so far, but not the bivalves. And they are likely bacterial mat eaters, so they'll be one of the
All right,
primary
let's come out.
consumers
Okay,
in this
coming out.
habitat. Of course, your primary producers would
Right
be
there.
the bacteria using the chemical energy
There we go.
via chemosynthesis
Okay.
to make food, similar to the
Is
way
there
plants
anything
on
else
land.
you want to zoom in to?
No, that's great. Thank you, Violet.
Okay. Gonna
Whereas
bring the camera up a little bit.
plants on land use photosynthesis to make organic matter from sunlight or light energy. And to eat those bacteria, then, we have our primary consumers, which would be the same as a cow or a rabbit or so on land. And that's likely what the same niche that those snails fall into in these habitats. They would graze on those bacterial mats growing there, and of course, as you can see, there's quite a lot of them, and they, given the plentiful energy from the hydrocarbon-filled seepage, they would likely have a large abundance of food. And hey, another one of these little patches with all these little Canoia miroglypta
like
snails,
there's a-
as well as a couple of the
I think loop in the lift line right there.
You know, it's
Is that
really
a salp or
extraordinary how
Loop
we'll
in the
come
lift
along
line?
and there'll be
Does that look like the lift line?
cold seep with a mussel bed, for example,
for it
and
to
the
heat.
tube worm,
Yeah.
What are you looking at?
or there'll be one
The
with the clams,
Sirius
and then
aft
here's
cam.
one
Okay.
with almost nothing but the snails.
I didn't see anything.
And here we've got another one of
but
these paraphila
I'll keep an eye.
Yep.
on the sea floor. Yeah, so cold seeps are really patchy environments. What is present will be dependent on the amount of flow in the area or seepage in the area, the types of fluid, being seeped, or gas coming from the sea floor in that area. Also dependent on the presence of other animals in some cases. So we discussed the ecosystem engineering by tube worms and mussels before, and the fact that they create habitat for other organisms. There are a lot of different factors, of course, other things like current, temperature. The seep environment, the chemosynthetic environments in the Gulf of Mexico tend to be dependent more on, or tend to be controlled more by depth rather than by geographic area. So in the shallower regions of the Gulf of Mexico, there seem to be very similar organisms.
Seen it now.
And then
Might
below
have been like a
1,000
siphonophore
meters,
just right next to the
there seem to
tether
be
that
another
looked
set
like
of
a,
organisms. The same general taxa,
you know, the
but
lift
different
line kind of getting
species.
pulled through one of the daisy
So it
chain
seems
loops
that
or
these
something,
animals
but maybe
are
it wasn't.
regulated somewhat by depth.
Keep an extra eye on it.
It looked like a little amphipod that drifted by a minute ago. And we saw another paraphylla bumping against the bottom.
Some more of these ophidiids, lots of little pock marks almost. in the
Yeah,
sea
they're
floor.
different. They're different than what we've seen. These little dark depressions.
Oh, getting into
Yep.
more areas
That
with
looks
more.
like it might be rock up ahead.
Have we seen this one before?
No, can't tell to the left.
Yeah, we have. Thank you.
Okay. Then to the left,
Certainly going up slope there. Looks like it's more irregular. Not rock yet. And our depth now is 794 meters. Quite a bit of a slope here.
Working in these geologically active areas is a little bit more exciting to me, because there's always this constant feeling that, you know, just around the corner there may be some kind of amazing chemosynthetic habitat waiting to be discovered. I guess that's the same with any type of exploration, and for any specialist of any kind, they may be waiting for some amazing coral colony around the corner or so on. But as I've said that, we are coming upon an area where it does look like there's a little bit more blackened sediment, a little bit more white bacterial mats.
I think another 8 to 10 meters in that move.
Copy.
Can we have another quick zoom, please?
Sure. Hold on video, let me just approach it
Some more clamshells.
from this side.
Okay.
And again, it looks like it's in one of these little depressions.
Alright, let's come in partially.
Okay, come in partial.
Hold right there.
Okay, holding.
Some more of those tiny little gastropod snails, bacterial mats. And although it doesn't look like there's much hair, as we've said previously, this sediment is likely laden with macrofauna and
Alright, come in a little bit more.
Okay, coming in.
There you go, hold right there.
Holding.
That's great, thank you. You can see how thick this bacterial mat
Thanks,
is,
Bridge.
how it almost looks like cobwebs coating the
Pilot's
sea
ship
floor.
is stopped.
Copy.
I'll get another one prepped.
Let me just bring it up a little bit. Okay.
Really beautiful. And it looks like that buccinid or Phymonorencus is dead. It looks like it's upside down.
Bridge,
no
RV
siphon
nav.
protruding. Some dead clamshells.
We're going to keep it moving. Another 50 meters, 50 meters, bearing
Dead
270
canoe
at
as
0.2 knots.
How do you spell that canoe?
Kanoia.
Good copy.
Canoe.
Okay,
Sounds
is that
like an
full zoom,
aftershave.
Lydia?
No, I have more.
Alright, hold on one second.
Okay.
Is that full?
There used to be one called canoe.
No,
that was spelled canoe.
Alright, there you go.
This is full. Also,
I
if you
never
look
knew
to
what
the
the
right,
right
there's a lot of things
pronunciation
swimming in the white bacterial mat area.
Alright, so come out a little bit. Look to the right.
To the left. Sorry. the like
that's
brighter
where those
patch.
snails
Okay.
are in the upper
There's a lot of
left.
things like tiny amphipods or something, like swimming just
Oh, yeah,
over
there's some
the
live
bright
ones of
area
those snails.
that we were just on.
Okay.
So,
Coming right there.
okay.
Right.
Yep, right there.
Yeah, I guess let's just come out.
Oh, come out? Okay.
Yeah.
Coming out.
Camera's at 55.
Okay.
Started another move at
So
about
snails in the genus
about 2 minutes ago.
Noe have been found in many
Okay.
organically enriched and chemosynthetic habitats, not just coal seeps, but also at food falls of various kinds. Some have been found from seeps, some have been found from wood falls. Many of these habitats do tend to share some of the higher taxa, not necessarily specific species, but genera and families will be shared between these different chemosynthetic habitats.
Are those canoia in their own family, or do you know what family they belong to?
I could tell you that, but I do not know offhand. Catigidae? Catigidae? That sounds right.
So that's a deep water family. I don't recognize that from shallow water at all. At least not in the Caribbean and Florida. So, did you mention Carolyn's comment?
Which comment is that?
We're just skirting the northern edge of the BOEM shallow gas seismic anomaly, and the GIS shows that we're out of the mapped anomaly area now.
Thanks, Carolyn. Another one of these paraphila. This may be the 6th or 7th one of this expedition, or not expedition, dive. And even if we are the ones causing
Let's
them
come in
to
partially.
Okay,
dive
coming in.
into the sea floor,
Right there.
it still means that there are quite a lot in this area. And we haven't really noticed, I think on one previous dive we may have seen some of these paraphila before, but as Scott had suggested very early on in the dive, perhaps there's some kind of spawning happening here, and that's why there are so many in this area.
All right, let's let it go.
Okay, coming out.
I'll tilt down a little for you. You're just on the bottom right, a little bit turned around. See ya.
Okay, thank you.
Yep. There you go.
And our depth is now 786 meters, so we are indeed moving up slope.
In the distance. Okay, we're going there.
There's a bunch of stuff.
So it looks like this may be our first outcrop of some kind, as well as a lovely little ctenophore there.
That looks like a burrowid. a carnivorous ctenophore that feeds on other ctenophores.
So this is really interesting. I don't know if any geologists are still on the line with us, but this looks like some kind of very regular feature. Perhaps just, I'm not sure if it's carbonate. But we can also see some
Do you want me to turn to keep you in view?
aggregation of dead chemosynthetic clamshells. So these are likely
Am I to your left?
You're off to the
and
right.
then we can see an
Off the right.
ancestor bivalve just in between the feature.
How far am I? Do I need to move?
I'm not
Those
sure yet.
are
I'd have to go over to check it out.
associated
Here, I'll show you.
with canyon environments and other deep
All right.
environments.
Let's come in partially. Video just a little bit so we take a look and then I'll get back in track.
Okay, that's partial.
All right.
That's
really
a real
bright.
pile. And there's at least one snail associated with it. I see a dead buck-sined at
All right, let's come
top
out.
left.
Okay, coming out.
I wonder if somebody dug those out and piled them up. What do you think?
I feel like no.
Okay.
I feel like maybe this is
It's a live
kind
one.
of like trap.
Yeah, there's something on the wall.
Yep.
Oh, look at that
was an
running
old area.
along
This
like
is so
that.
Weird. So can we just stay here for a little bit, and we want to
Yep.
get some really
Are we
good
moving?
zooms
I
of a lot
think
of
we're
this
almost
stuff.
done.
Okay.
A lot of these walls are
Good.
covered in
Especially-
animals.
Right.
Stop it.
Okay, yeah, we'll do it. Bridge, RV Nav. Can we please bring the ship to an easy stop here?
So we'd really like some.
I realized
good
your head
views
was
of
all the way over.
Of the rock when we can, as well as some of these animals inhabiting it, please.
Copy.
Oh, there's a whole bunch of the Acestas along this low edge here on left of center, and all along the side here, and then the great big one.
All right,
And
let's
then
come in,
another
video.
Rockinia.
Okay, coming
Thanks,
in.
Bridge.
On
Spider
the
crab.
All the little
Yeah, sure.
ones are assessors.
Oh,
I think
okay.
they're brachiopods.
Can
Oh,
we
yes,
move,
they are.
like,
Look at that. They are Brachiopods.
5 meters toward you2.
See if they
And,
want to adventure down it.
In case you
Okay.
I
need.
think we're okay where
I
we
don't
are
know if this
otherwise.
is- no,
All
that's not Rockinia.
I think the crab has something in its claw.
Its
Can I come in?
Yeah, sure.
rostrum. It looks more like.
Oh,
Oh,
brilliant. That's
Uh,
great.
well,
Keep them there,
Or,
please.
I guess
got
it may
things
have.
hanging.
Yes, it does have 2 prongs. They look broken off, so it could be a Rockinia. There's a little zoanthid growing on it. So these clam-like things are brachiopods, which are commonly called lamp shells, although they don't look like, bedside lamps or anything like that.
Let's come out a
Lamps
little
that you
bit.
found
Okay,
in your
coming out.
home.
There you go.
There's one kind that, if you look at it inside view, looks like the kind of oil lamp that was used in ancient times, and you'd rub and genie would come out of. So these guys, they do have 2 valves closing like a clam, but the valves are arranged differently. So in a clam, the hinge is dorsal, and the 2 valves are left and right. And in the brachiopods, one valve is ventral and the other is dorsal. So the orientation is completely different, and so is the anatomy, development, and everything else. These brachiopods have a horseshoe-shaped ridge on their internal visceral mass, and that ridge sports a line of ciliated tentacles. Those can be elaborated into coils, and like many other deep-sea organisms with a large surface area of tentacles or filaments or projections, they are suspension feeders. And the cilia on those long tentacles filter this, create a current, bring water and suspended particles into the brachiopod on which it feeds, and of course brings oxygenated water as well.
All right.
So the
Video, do you
structure that
wanna
ridge
zoom in
like
anything particular?
ciliated,
those like clear
the ridge with
white
the ciliated tentacles
creatures.
Along
Okay.
the feature.
is called a lophir.
All right, let's come in.
And
Go in.
that
Oh,
puts
do you
these
mean the ones
brachiopods
on
in
the crab
same
or
group
down
No,
there?
no,
Down on the rock?
as
on the
another
rock.
group we
Okay,
saw
let's come out.
a number
Okay,
of
coming
dives
out.
ago called the bryozoans.
All right.
which are colonial.
And then,
what do you think, Jeff? Do you want to move?
Let's
or
go in.
comfortable
Okay,
here
coming
still?
in.
Colony are much
Still
smaller
looking okay to me.
than
I mean,
these
Right there.
brachiopods.
Okay, that's
You wanna
full
move,
And
zoom.
we
each
can.
of them
Copy.
have a
not really pulling on them or
a
anything.
lophophore with ciliated
got a good
tentacles as
catenary
well. And there's
tether and
some
everything
crusted
and all.
sponge.
All right, we'll leave it here
There
for now.
are little polyps there on the rocks which look like
Yeah,
zoanthid
it
anemones.
looks okay to me, but
And the
happy to
little
move if you want to.
fuzzy brown trees
See where
are
Fernando
agglutinating
ends up going.
foraminiferans,
All right, let me know when you're good, video.
which
Okay.
we've seen on pretty much
I'm-
every habitat.
I'm good.
All right,
It
let's
depends
come
on
out.
what science wants to focus on.
Watch lead, do you need anything else?
Yeah,
Do you
so if
wanna...
we could get some looks along the feature, so like first in between these two, in between the gap there, we'd like to take a look at some of those animals,
right.
as well as then move to the right
Okay.
along the feature, please.
To the right, so I'm probably gonna be moving that way.
yeah, okay. So maybe we do want 10 meters
All right,
to get us
let's-
along there, or
Just-
5 meters.
Just want to do 010.
Let's come in.
Okay, coming in.
Yeah, 010.
Hold it right there.
So
Holding.
there's a nice buccinid gastropod
and give it
and
10.
what looks like a tiny little pink,
Thanks.
possibly galatheid. The big
Bridge, RV nav.
clam-like
Like a move,
bivalve
please,
is
of
indeed a
10
bivalve.
meters
It's in the
at 010
genus Acesta,
at 0.2
and
knots.
it is a relative. It's a file clam related to the scallops and thorny oysters. So very completely different anatomically from the brachiopods,
Good
but
copy.
doing something very similar.
And these Acesta tend to be found attached to hard substrate, usually in like canyons and so on. So Carolyn Ruppel, who's joining us from the from USGS, she seems to think that this feature we're looking at is
All right,
a
video. Is that full
carbonate-led
zoom?
outcrop.
No, I have more.
And she says
If
it's
you want,
very
you can come in.
pitted
There you go.
Okay.
with lots of erosional features. That's great, Pilot. Thank you.
Okay, whenever you're good, video.
Okay, just a couple more seconds.
Copy. Holding.
And we're good.
All right, let's
Okay.
come out.
Okay, coming out.
Thanks, Bridge.
So Acesta
All right.
have this
That's full
very
live.
oval
Losing
shape
skids.
and have this characteristic orange, very orange insides.
And they are attaching by threads like a mussel does. This is
All right, let's come in
Abyssal
partially just to
threads,
look at
and,
the rock.
some of their
Okay,
shallow
coming in
water
partially.
relatives are free
Right there, that's
swimming,
good.
like some of the scallops.
This is very interesting. Was not expecting this at all. And it's interesting that there are no corals in this area. You would think that this would be enough hard substrate for there to be corals or
Let's
crinoids
come
or
on the
something like that, but all we have are
on
these
the conch right there.
crusting
Let's come in.
sponges
The conch.
and
Yep,
brachiopods
right there.
and
Hold it.
bivalves.
There you go.
Another one of these gastropods, either
Yes.
buccinid or Phymenorhynchus. Some squat lobsters below.
All right, let me know when you're good,
A
video.
live
Okay,
cup
I'm good.
coral,
All right,
which is
let's
there.
come out.
Okay.
Yep.
Up in the upper right, there's like a
Maybe
anemone or
Desmophyllum?
something, like
Up
sticking out of the.
Right.
It kind of looks like a cup a little bit, but it's in the sediment.
The sediment. Oh, okay.
Between
I
the
see.
two.
All right.
Yeah.
Let's hold on to that.
Scleractinian coral, that would be cool. Since we haven't seen any coral yet.
All right. Let's try to come. Hold on. Let's come in.
Okay, coming in.
Hold right there, please.
Holding.
All right.
Yeah, there's a couple of them. 2 scleractinian
All right.
singles.
Do you still have
Single
quite
Yes,
a bit?
I
All right,
do.
Hold on one second.
Okay.
All right, let's come in.
There's a nice
Okay,
little
coming in.
cup coral, couple
Right.
of them.
That's full zoom.
Copy. I'll try to hold that.
I don't know them well enough. I don't think that's Desmophyllum because it's not oval, really oval like the ones we saw the other day. But there's a whole host of them in this part of the world.
Thanks, Bridge.
It's great, Pilot. Thank you.
Whenever you're good, video.
Okay, coming out.
Copy.
Turns out that of the almost
All right, anything else you see on the shot video?
of stony corals that we usually think, of course,
I don't think so. I think
are
everything
the
else
reef
looks
builders, the colonial
similar.
forms with the symbiotic
Okay.
algae.
Plenty of space to
And of
move
course
down
there
now
are
along it.
hundreds
All
of
right.
species on reefs in the
Let's
Pacific.
move
And,
along though.
maybe 60 or 70 species in the Caribbean and Florida, but fully half of all of the stony corals in the world are solitary cup corals like that, and the majority of them live in the deep sea.
If that one's loose, if they'd want it.
Do you want any collections here, watch lead, or just having just moving along it is enough for you?
Why not? You can find a piece of the rock.
Right between your skids now is what looked like a little loose piece, but I don't know if it was good enough.
Something might be lying loose on the surface there.
Oh, so what about this?
Yeah.
That little piece right by your right skid.
Okay.
And it has a brachiopod on it.
Oh, that'll be a bonus.
Yeah.
I'm gonna turn my head a little bit to get you again.
Not.
Copy.
After that move, just gonna line up northeast.
And just in the distance, we can see more areas of reduced sediment and bacterial mats.
There you go.
Come on, land.
That will be cool. The I know that there was a
turning on lasers
an
for a
article,
bit. Copy.
A major article on brachiopods from the Caribbean Sea and adjacent waters that was published by Cooper back in 1977, but that would have likely been based entirely on
this is going to be a little crumbly and has stuff on it, so we'll probably put it in a bio box.
Much in the way of
double check
submersibles,
with the
although I
watch
haven't
leads.
read it.
Okay. I don't know where to, because I think I have something on my starboard and I can't land.
What's wrong? Oh, you're sliding?
Yeah.
You might want to do a little bit of, you know, this is the downward slope.
Yeah.
So you want to push into the slope and then only push down
switch
with,
over to
you know,
camera
10 or
2,
15%.
which is the Sirius view.
You don't want to push down all that hard.
Sense of how big this feature is
Right there, I'm too far, right? Yeah, or I'm not that far.
No, I think that's hard to tell. I think you can reach from there with the nip.
Okay, let's try it now.
Put in your auto heading as well.
So we completely forgot about sampling today until our pilot, Fernando, reminded us. On the Okeanos Explorer, we tend to try and balance 3 objectives. They are exploring, getting great video.
It's going to be at the very end of your reach.
Might
be close enough. It's hard to
well
tell.
That's most of our data.
You're going for the one
as well
where the lasers are now, or one to the right?
The one to the right. So having to
give it a try. You
all
might
of
have to
those
push forward a little
means
bit.
that we have limited time for many of them. So we tend to only like to collect a maximum of 6 samples per
It's probably
dive.
within reach.
Those have to be shared
let's try
between
it.
geological and biological samples.
Make sure you can get it set up. Make sure you once
we
you
have
start the
certain criteria
hydraulics,
for those collections.
check your
So
grip force. Make
they
sure
have
it's
to
either
be
1 or
either
2 with this
something-
kind of rock.
An animal that is poorly known,
Anything higher will
or an
likely
animal
crush
that
it.
is rare,
All right.
or an animal that is a new
You're totally
record
spun
for an
up.
area,
And
or perhaps
you
something
might want
that
to
is
check
even
with
a new
watch lead which
species.
box you want this to go
Yeah.
into, in case you want to open
Additionally,
the
we can
port
also
side
collect
rock
something
box
that's very
before you pick it up.
So watch lead,
of the area.
I was thinking about collecting the one to the right of the lasers, the one that's out there by
The
itself.
smaller of the yeah, that looks great because it has a
Don't see any other samples here.
brachiopod on it as well.
Would you like that to go in the bio box or the rock box?
If possible, a bio box would actually be better.
Okay.
So yeah, so we also can collect things which are more characteristic of the area. And we also collect rock samples, geological samples, which will help us to understand the origin of this area, as well as many of the geological activities and characteristics of this area.
So,
and so by
you'll
collecting
power it up
something
first.
like this, I know Carolyn was saying that will they be able
Perfect.
to date
And
this
now if
carbon
you see there's
and
a
get
grip
sense for
force.
when this actual
2. So
feature
2 will
was
close
created?
fairly slow, but you'll still have
Another
enough force
parafiller
to
drifting
give that a
into
little
view.
bit of a squeeze, so you don't want to clamp
But
all the
usually
way
we
down
put
on
our
it.
rocks in the rock box,
Grip force
which is an
1 will
open
slow,
box on
close
our
really,
ROV,
really
but
slowly,
because
but
there's
guarantee you don't
fauna
crush it.
attached to it,
Okay, so should
we
I
can
just
see
leave it
some
on 2 or should
brachiopods. We're
I?
actually
I would
going to put
recommend
it
2.
into
Alright,
our
indexing
bio
in.
box, which has a lid, and
Arms live.
as a result, the temperature of the water remains pretty stable.
Hopefully I can reach.
Yeah, go in with the closed jaws and give it just the slightest poke first to make sure it's loose.
Okay.
From one side or the other, just a little nudge.
Jaw lock is the one on the right, correct?
It's on the left.
On the left.
Yep.
Okay.
There you go. Yeah, and the index sometimes doesn't work, so make sure the light is lit up when it's not running. Oh, definitely loose.
Yeah, it is loose.
Cool.
Alright, so let's open slowly.
Nice. Good control.
I think
Would you like your mini Zeus on the main screen here?
Sure.
There's your mini Zeus view. I'm gonna line it up a little better. You can always take a zoom on the Zeus if you need a little bit better.
Yeah, let's come in on Zeus
there.
a little bit.
Okay. On mini Zeus?
Yeah.
Okay. Coming in.
See, you're quite a ways. See how your wrist is coming up? You're quite a ways behind it now.
Yeah.
Or in front of it. Or
That's a partial zoom, pilot.
Thank you. So let's just go. I keep on. Which one is that?
Are you inversed? There you go. Yeah, pull that towards
I'm
you.
going to be interested to see what brachiopod this turns out to be. I'm looking at the
yeah, you may.
section on
Yeah, I'm
brachiopods
inverse,
in the
so
Gulf of
that's
Mexico
kinda weird.
origin waters and
What's wrong?
the biodiversity.
So like if I go like this, it's actually
component.
pushing away. You
And
see?
Yeah, that's
there
how it should
is
be.
That is how it
Biv-
should be.
Yeah, because you're fully
a
extended.
Okay.
It is actually
brachiopod that is
that's
known from
the right
between
direction.
about 700
Alright.
900
Yeah,
meters.
there you go.
That's
Called
kind of strange.
Ecnomosia gerda, probably because it was first collected by the University of Miami's research vessel Gerda.
There you go.
I'm going to see if there are any others listed at that depth range. Another one called Platidia anomioides from 700 to 1400 meters.
Yeah.
Ticosina elongata from 730
Straight up with the
meters.
shoulder will get you out of that
So
dirt
there's
quickest.
probably a limited number that this could
or
be.
with the elbow, either one. But
And we'll just
there
have to
you go.
send it to
Nice
a
work.
brachiopod expert, or have a
Ooh, that's
brachiopod expert
big. That was
requested
deep.
on loan
Look at that.
from the Smithsonian.
All right, let's come up, video.
Okay.
I'll follow you
Art
with
on
the
ROV HD2.
That's a nice sample.
follow you with both here, or with the HD2. There, you can just see you on the top of the screen and where you're headed here.
All right.
Very
I assume you're
muddy
going port side, right?
because it was
Yeah.
buried partly
That's
in mud.
full wide on mini Zeus, pilot.
Copy.
Be interesting to see, in addition to, yes,
See your
lot of
minute
mud
cam, see
falling
how low
off
you are.
Yeah.
On your
Before we
starboard
put it
minute.
in, if we could
So I'll pick it up
get
a little higher. There you
zoom
go.
of it, do a little twirl.
There you go. Ready for me to open the drawer?
Pilot, they want to zoom on it.
Okay. Oh wow.
We figured it was a little bit fragile, and
Go ahead.
we were kind of afraid of
Go ahead and zoom in.
it breaking on us.
Okay, coming in.
Valves on
Looks
there.
like there's quite a lot of sediment holding on there.
right, and it looks like it's made up of these knobs and sort of finger-like structures.
So if I press this one, this is to actually rotate it, right?
Be interesting
No,
to
this
see
one
what
is-
else
Yeah,
is on it
this
besides the
this
Brachiopods.
trigger is, yeah. If you hit that button,
Looks like there might be
drop
encrusting
it.
sponge.
Actually, wait, so the way you've got it set right now, when you hit this button, you're gonna drop the redepress. That should be lit up. Yeah.
I'm not
I
letting
missed
go.
it, but Tina has
sure.
said
If
in
you-
the chat room she
yeah, there
saw
you go.
some Ophioids
Yeah, so you have to
fall off.
index out just to rotate. There you go.
Yeah, there's a lot of sediment there.
You can see how sticky it is.
All right. So let's come up,
Okay,
Edie.
coming out. That's full wide.
All right. And then, do you think you can open the?
Yeah. Yep. I should have room to open the box without any issues.
right.
It's going to be a messy time in the wet lab this evening.
Okay, looks like you're pretty much over where the box was going to be, so you want me to open?
Yeah, go ahead.
Hold. Okay, here we go. Extending drawer. Getting a little bit close, but I think that's going to be enough room for you, right? I'll tilt down a little more on the Zeus, on the mini Zeus for you. I can open it a little bit further if you need a little more space to get it in there. Just let me know.
I think it should be okay.
Okay. Nice work. Now just careful as you open the jaws and pick up that you don't punch the manip cam there.
Okay.
It'll be open pretty wide. So just raise up a little bit away from the rock and then reclose them. Might have to give a little shake. It looks like you're stuck to it. There it goes. There you go.
Excellent.
Perfect.
All right. Oh, hold on 1 second.
It's okay. Just the elbow towards you. The wrist towards you, I mean. Shoulder down a smidge and wrist towards you, or wrist up. You're just hung on the bottom of that box. So shoulder down will take the finger off the box.
Okay.
Ledge. Oh, like that. That was a fair amount of force.
It looks like the fingers are maybe hitting hard on the divider in the bio box.
Okay.
Right there, I'm not indexed in, right?
Yeah, what's your force feedback?
You have
at?
a little bit
3.
Sorry.
This one?
Yeah, just making sure it's on. What are you caught on? What's he caught on?
I was thinking maybe the fingers are scraping alongside the side of the bio box and getting caught on that.
Yeah.
On that lip.
Just try this joint here. Oh, you're indexed out.
Yeah,
but when
I'm
you index in, just pull up on that joint and see if it will rise.
No, it's not rising.
Yeah, you've still got this inversed. Your wrist up, down, you still have inversed. So you pull it, when you index in, pull it towards you because you're jammed against the inside of that box.
So
pull it towards you.
It
This
should
way.
go like this.
This
Right.
part should go towards you a little.
All right.
And then you're going to have to pick up sort of at the same time. Because you're really cramming it against the box there.
So, hold on, I'm gonna index out. So if I go like this, just like this, by
Yep.
itself,
Yep.
it should go like this, right?
It should, yes. Yep.
So if I, okay, so I'm just gonna lift it a little bit like this and just come like this.
Exactly.
All right.
Yep.
So I'm gonna try that. Indexed in, and then lifting.
You might be pushed hard against the bottom, which is why you're not having much luck. So.
sure it's this way? It's not that one?
Yeah.
It's not moving at all.
Yeah.
I think it's...
It's just hung up. Sometimes when it gets hung up like this, that'll happen.
Alright.
Are the jaws opened or closed right now?
They're closed.
Closed, okay.
Yeah, I haven't let go of the trigger. It's fully closed.
Why don't you just put
twisting
grip
the
lock in?
wrist.
You put grip lock in, and then you don't have to worry about depressing the trigger.
So grip lock in, right?
This one's grip lock.
Oh.
That's wrist, yep.
Okay.
And yeah, try turning your wrist. Try some things. It looks like you're a little wedged.
Alright, so I'm gonna index in.
There you go. Yeah, so you're able to do that.
I can move the wrist.
Yeah, so then lifting just this joint up.
See it scraping the inside of the bio box wall, and I think it's just a lot of friction there as you're trying to pull up. It might be getting caught on the lip.
This one.
I think maybe you should elbow.
Could try elbowing
up a little bit.
He's maxed out on the elbow.
Is it?
It's the wrist that is,
Okay.
and the wrist is jammed in, and it's not. I see him lifting up. It's not extending for some reason.
Yeah, I cannot do this. I'm trying, but
yeah, I can't.
Alright, I'm indexing. I mean,
yeah, just isolate it. Perfect. Yeah.
So I want to do
this. Yeah, tap that and try it. I see you trying it. I see it not working.
Alright, so I'm indexed in, and it's, everything's hopefully isolated. I'm trying to just move the...
What if... Oh,
there
we
it
go.
goes. Yeah,
Okay.
there you are. Perfect.
Alright.
That's what we're looking for. There you go.
Alright.
So now,
just
elbow or shoulder straight up. The shoulder. So just pull this towards you, and keep everything else totally stationary, and just pull it towards you, and it'll all lift up in a straight pattern. There you go, just like that.
There you go.
Boom. Clear the box. I'm gonna retract.
That's... Okay.
Yeah, you got yourself turned around on the wrist. That's tricky.
Yeah. Now what?
Now your wrist is still tucked in, so you're gonna have to pick it way up to stow, and then you're home free.
All right. Can you bring the...
You got the mini Zeus
the
up.
mini
so you
Zeus
see?
this way, yeah.
Just careful how much swinging around you do up high, because there are cameras you could whack up there. There you go. Perfect. Exactly right.
Yeah, that...
Awesome. There you go. Home free.
That was a little bit harder than what it should have been.
So do you think was the wrist pitch not functioning properly, or was it just that there was so much friction?
I think it was just hard against the box.
Against the box.
Yeah.
Okay.
But it was against the bottom of the box? Or why
I think
wasn't
you
it?
did get yourself wedged in the corner.
Yeah.
So it wasn't until you raised up a little that you gave yourself the room to extend.
All right.
Get you your slim camera back.
Okay.
And watch, this is Spec 01 Geo, correct?
Correct.
Okay.
That was... okay, watch lead. I'm back with you.
Sounds good. Thank you very much for collecting that carbonate for us, as well as whatever animals may be inhabiting it. We're very excited, and in fact, a lot of the scientists in the chat said that folks following along from England, British geologists, were very excited to see that rock getting collected because they intend on dating it. So thanks very much from them. So we are going to continue along this feature. Can we get a quick snap zoom on that urchin?
On that what?
The sea urchin, just to the left of the feature, the spiky animal, just to the left of it.
Is that an urchin?
Right
I
by
think
the-
it's
underneath the ledge. I see it.
Oh, okay, I see it. There you go. Let me just come this way.
So just so you know, you put uphill behind you. So when you go to land, your tail's going to land. It's going to be a little funky, but you can do it.
Okay.
Doesn't have to be a long look, just a quick snap.
Okay, let's come in a little bit video.
Okay, coming in.
There you go, hold right there.
Brachiopods alexandri,
Yeah, that's what I would have said.
and lots of the brachiopods.
It's beautiful. Thank you, pilot.
All right, let's come out video.
Okay, coming out.
And is that a different crab? It's a little pinker.
Yeah, can we have a quick snap zoom there on the spot lobster?
Thank you, video.
Yeah, they. Oh, I think that they wanted a snap zoom on that squat lobster that we just went by.
All right, let me just bring it back.
Getting kind of high, just so you
Yeah.
know.
Yeah.
I'll come up a little for you.
Okay.
It's going to disappear under a rock.
All right, let's come in.
Okay, coming in.
Yep. It looks a little different than the ones we've seen on a regular basis, isn't it? It's not Emunida, but it's redder than the Munidopsis that we've seen. I'm not that familiar with them.
Do you want me to come back out?
Some solitary cup corals among
Here,
the
I'll come full wide.
brachiopods.
There you go.
Okay. Yeah. Is that okay, watch lead, or do you want to...
No, that's great, thank you. We can continue moving up slope.
Okay. Let's just come out video. I'll just
Okay.
bring the camera up.
Okay, that's full wide.
All right, thank you.
Okay, can I lose skids, pilot?
Yep.
Okay, we can line up back towards the west, I think, unless you want to follow this outcrop more.
All right.
Today.
I think she said uphill, so I think we can continue on towards the west to waypoint 2. So I'm facing pretty much northeast right now, so we need to pretty much spin a circle. I'll
Okay.
stir it around a little bit here,
Andrea Quattrini has
but it's going to be almost like you're
says
flying
that
under me.
the
All right.
Or you can
chimera
or you
that
can
we saw
lateral over
is
if you want.
Hydrolagus mirabilis.
There, I'll hang out here. This
She
is
spotted
northwest.
some distinctive characteristics such as the indentation of the second dorsal fin, which
Coming
is
around as you
diagnostic.
do.
More of those paraphila going by
Yes.
as well.
And
Too far.
Marsh Youngblood has
I'm pretty much
said,
faced west. You're
the
right there in my
abundance
bottom right
of
corner.
that
All right.
Gotta
Medusa
pop into view
species,
any second now.
paraphila, that
There you are.
conical purple one, in water at this depth may be related to the relative shallow depth of the sea floor here, because
go. There's
it
west.
often lives deeper
Okay,
in the
thank
water
you.
Let's
column
get a little
than
push out. We
1,000
can get
meters.
our moves going again.
And they're vertical migrators.
Sounds good.
So as they drift along with the currents and migrate vertically, as they go down at dawn, if they encounter water, then water that is shallower, or the sea floor that is shallower than they want, they're going to basically come up against it. And, of course, if they survive, they'll migrate up at dusk, and hopefully for them be...
Oh, is this video?
Hopefully
coming
be
partially?
carried elsewhere where the water is deeper for their next migration.
No, I don't think that's anything.
Thanks,
really.
Marsh.
More bacterial mats.
All right,
which is
let's come out.
Okay, coming out. Closing skids. Well,
Copy.
You're out there pretty far. We can do a move due west.
Andrea
15 meters
Quattrini
or something.
also threw out the possibility that Chimera might possibly be a new species.
Yes, can we get a move, please?
That's always a possibility.
50 meters, bearing 270 degrees, 0.2.
Alright, let's come in right here, video.
Okay.
Partially.
Coming in.
Let's see what this
partially.
is.
That thing at the bottom?
Yeah, let me just bring the camera down.
That looks like one
Good
of the
cover
snails.
bridge, thank you.
Go ahead.
Okay, coming in.
Right there, hold.
Holding.
So that looks like a piece of Sargassum.
And here we have a snail plowing through the sediment. And so that is the
upper left
culprit
pilot.
Alright, moving
behind
there.
some of these narrow trails that we've been seeing. And it
Oh.
looked like it was crawling along, buried itself, and then came out of the burrow again.
Alright, it's moving slowly.
Unless there was,
Good cover
well,
bridge,
no,
thank
that
you.
could be a little burrow that was there.
Can I come in,
and it
pilot?
looked like the snail just made a hard left turn past that burrow. And it looks like it's got maybe hydroids growing on it, or agglutinating foraminiferans.
Is that full zoom?
Not sure what those are. Those little
that
tubes.
is.
Okay.
Or filaments.
Let's come out a little bit.
Okay, coming out. Just a little.
Yeah, thank you.
I think we're getting closer.
Yeah, I'll just land right here.
Oh, is that a octopus in the Seriosphere? Right there,
Yeah, it's been hanging out with us for a while.
isn't it?
I can zoom in on it.
I thought it was a
Or a squid.
Or a squid.
Yeah, I thought it was a Holothurian.
yeah.
But
I thought it was a Holothurian as well. I ignored it.
That could be a I can't tell what that is.
I can zoom in on it, co-pilot.
It's not really staying in view
Bob
very
Yeah,
long.
it's not.
I don't think it's an Hypniastes or another Holothurian. Looks like it's got an eye on the
I'll
right
just come
side.
up a little bit. A smidge so you guys go up out of the light pool as much. Down.
Unfortunately, this is a mollusk off here. We have a beautiful gastropod on the screen, but then on the other screen there is a potential cephalopod.
Yeah, you can come in partial so you can get more
Everyone's
detail.
eyes are on the cephalopod. Oh, nope, a Hypniastes
Yep.
fooled us.
I was going to say,
Shallower
my
than
vote's on
usual.
Holothurian.
Yep.
Mhm.
And Andrea
Okay, coming
just
out
told us
on
on
Sirius.
the chat line that
That's full wide, co-pilot.
David Ebert, a
Perfect.
Chimera expert, confirmed that the identity of that last one that we saw, last couple that we saw, was Hydrolagus
Alright, let's come out
mirabilis.
on main
He
Zeus.
said it was a neonate.
Okay, coming out on
a
RVHD.
youngster,
There you go, thank you.
and they can be tough to identify.
Okay,
Interesting.
gonna zoom past skids.
Thank you
Copy.
guys very much.
Bring camera
And
up
so
a little
that's
bit.
a
Okay,
great
sounds good.
example of telepresence. We're working with two ichthyologists on shore, Andrea and Ken, while Chuck and I know very little about deep sea fish. And they are then able to reach out to other colleagues around the country. David Ebert is a Chondrichthyan deep sea expert, and get potential advice from him on the matter, on what we're seeing in near real time.
do you want to sit here?
In 10 minutes.
Oh,
I
in
will.
10 minutes.
Yeah.
Okay. All right.
Yeah. The watch changes then.
All right.
Ooh. Can we go have a look in this depression, please, pilot? To the left, where
to
there
the
are all
left.
those things shining?
Yeah, I don't know if I have enough tether, but I think we are moving in that direction, so I'll get there in a little bit.
You have room to get out there. I don't see the second
So
football
we're
yet,
about
so you can push a little ways.
150 meters
All right.
There it is.
from where we,
got a little ways.
the top of the ridges. We were hoping to get to some hard substrate at the top of that ridge, but as we progress up there, we are seeing areas that appear to be more active than others. And this one looks like once upon a time, there was a lot more seepage than there currently is. We've got some bacterial mats, some more dead clamshells. Ooh, another one of these crabs emerging from the sediment.
With a very large claw. Look at how big that claw is.
Let's try to get the crab. Hold on 1 second.
Okay.
Let's come in partially.
Okay, coming in partial.
All right, hold on right there. Where did it go?
Should I come out, pilot?
Yeah.
Okay,
yeah,
coming
let's
out.
come on. It's hard to just stay when you're going downhill. Hard to get those shots.
Yeah.
Okay, so let's try to hover right here. Let's come in.
Okay, coming in.
Right there, hold
I
right
don't
there.
know.
There you
I think
go.
he's challenging you.
It looks, again, spider crab is as close as I can get on this. It's very different from Rokinia with that enormous claw.
It's taking us in, sussing us out.
I have more
Having
zoom
a stare
pilot.
off with you two.
Yeah, come in.
Okay, coming in. See if we can see his face.
Looks like he has
Face.
not
Okay, let's try to go for the
shed that
face.
exoskeleton in a while. It's all overgrown.
Oh, I see an eye.
The bushy little trees look like
we're getting
glucinating
closer.
foraminiferans. I'm not
Yeah.
sure what those on the top are. They might be also foraminiferans or some kind of hydroid.
Oh. I'm going to come out partially,
Yeah,
pilot.
there you go.
There.
Yeah, let's try to
That
hover right
works.
there. Better.
Wow.
So Bob Carney
He's got quite
said
the ghillie
that
suit.
this crab is Trichopeltarion.
All right, maybe you can
which
come in
is
right now.
also
I'm pretty
found in
stable.
Okay,
mussel
coming in.
beds. Tiny little hatchet fish there.
He looks surprised.
Great, Bob. Thank you very much. Look at that detail.
Is that full zoom?
I have a little bit more, I think.
You can do what do you want?
Okay. Okay, I'll come in just a little. Oh wait, no, never mind. That is full zoom.
Okay. Whenever you're good, we can come out.
Nice. I like the composition. Okay. Great.
All right. Got you 60 degrees, a little bit off to the right there.
Copy.
And my heading is
Uh,
starting to run away from the west, so I'll actually
Do you wanna
10-ish degrees.
Do you wanna quick zoom of any of this? Watch lead.
Yeah, sure. I mean, if we could zoom right here in the center again, it looks like more of the.
All right.
bacterial
Let's come in right there.
Okay, coming in.
Coccigina, or
Right there,
some
hold.
kind of a
Holding.
sycamide dead. You can see how thick that bacterial mass looks there, and also
Come in.
some more of these
Coming in.
Pnoye Muroglyptas. These
Right there. Let's try to.
reducing habitat.
There you go.
specialists.
75 degrees.
Copy. All right. Let me know when you're good. Video.
Yeah.
Boy, that is a thick bacterial
Just
mat.
a couple
Look at
more
that.
seconds.
And then there are also what look like maybe tiny amphipods. Or
Yes, that's right.
I'm good, but science is
Unless
still.
they're tiny gastropods.
oh, and there's also a scale worm just there on
Yep.
that shell,
Yep.
which is a type of
Couple
polyp.
more seconds, pilot.
All
If
right.
these look like little organisms. These
Let me know
little
when.
Okay, I'm good,
elongated
pilot.
pale
Co-pilot, we're
creatures,
good.
but they're not
Let's go.
moving.
All
like
right,
amphibians
let's come
just
up.
about
do.
complete.
They might
Thank
be tiny
you.
Thanks.
gastropods.
Okay, that's full wide pilot.
All right, lifting off.
Okay.
Bringing camera up.
Okay, I'll lose skids after you bring it up.
Right there.
Sounds good. There we go.
All right, moving in, moving up.
See you.
Okay.
Thank you, bridge. Move complete.
Copy nav.
So this once would have been a very large clam bed.
Don't expect it to be steep, but it's definitely steepening. So maybe the next one should be
But
30.
now is no longer.
How far do we have to waypoint 2? Generally.
And another
140.
Solo.
Okay.
Couple
Two of them.
Yep.
Yeah.
Yep. Possibly a halosaur there or synaphobranchid, couldn't quite tell.
Coming partially to see what's this. I think it's
Okay.
another shell.
Yeah, looks
think
like another
it's
shell with
shell
some
sargassum.
and depression.
All right, we can come up.
Okay, coming out.
Bringing camera up.
Delta 15,
And losing the skids. There we go.
camera angle 45, you're just at the bottom.
Okay.
Ready to keep going?
Yeah, I can push out. There's not a lot to see here.
Yeah, let's do 30.
Bridge, ROV nav. Yes, another ship move, please. 30 meters, bearing 270 degrees, 0.2 knots.
What's that little circle?
What is that?
It's probably garbage. You're good.
Good copy, Bridge, thank you.
So we're working our way up the slope towards waypoint 2.
There's a thing like swimming towards you.
Bathynomus is swimming above us.
Oh,
It's to your starboard, yeah.
Oh,
There it is.
What is it?
Away he
Oh, I
goes.
see.
Oh, but he really doesn't
Oh.
want to get. Yeah, I don't know where he went now.
Dive bomb.
Thank you, Bridge.
And look at these sort of areas of all of these little irregular sort of pock marks in the sediment running irregularly along the same portion of the slope off in the distance, and then it becomes completely smooth again.
Yeah, so they could have been areas where liquid was seeping out of the seafloor, this hydrocarbon-rich fluid, and that would have eaten away little pockmarky areas or even gas at one point. Maybe there'll be more Bathynomus giganteus when we get to the top of the hill.
Look at that little thing flap.
Oh, can we get a quick zoom on this?
Partially video.
Okay, coming in.
That looks like a pteropod.
Right there, hold.
Black pteropod or...
Damn.
That's weird. Looks like the wings are coming out of the sides of the body instead of anteriorly.
Okay, you can come in a little bit.
Okay, coming in. Oh,
Let me just tilt up.
okay.
Can you tell if it's coming closer or?
It came
going
closer
away.
and then now it's going away.
Okay.
I'm zooming out just a little bit more, keep it in view.
All right. Let's come in.
Okay, coming in.
Tina asks,
There you go.
"Are
Hold
you
it
able
there.
to see siphons
Holding.
of the
Tether's
clamp
getting
sticking
awfully
out
close
of
to
the
your
mud?"
upper swing
So
arm.
we
Just be
had
careful.
varying opinions on this last time.
Yeah, Roger.
That's weird.
It's getting
I
closer.
have seen them
All right.
of various
All right, thank you.
Vesicomyines
Try to
before.
hold-
It's just peeking out of the floor.
Yeah,
I'm
let's just
zooming out.
let it go.
But
Okay.
someone
It's
else,
okay.
I can't remember who, I'm very sorry, maybe Eric Cortez was suggesting that in some places they may be much deeper down and you wouldn't be able to see their siphons. So, mixed views.
Can I lose skids, pilot?
Yeah, go ahead.
It's curious that we've not seen anybody at the entrance.
Pretty
to
far
those
off to the
tunnel
right
burrows.
there, pilot. Just so you know.
Another crustacean in the distance, scooting along the bottom. That may be that long-legged hermit crab that we saw earlier. Can't tell.
Yep, we've
Yep.
definitely seen that before. That very characteristic movement, which for folks who don't like crabs and spiders may be a little bit creepy. Can we get a quick zoom on this? Yeah.
Let's come in, video.
Okay, coming in.
Hold on, let's come out because
Oh,
I
coming
think
out.
I'm past.
That looks like an anemone of some
There
sort.
you go.
Okay, coming in.
There you go. Let me just bring
Quite
down the
tall
camera a little
black.
bit. I think that's.
I don't see an inner ring of tentacles, but it may be too small.
We have not seen anything like that for this expedition.
Nope.
All right, let's come out and I'll try to land.
Okay, coming out.
Still trying to puzzle out what that little dark maroon swimmer with the wings was.
Well, what else could it be?
Well, you know, a gastropod of some sort, but
All right,
that
let's come
tail
in.
flap.
Okay,
I've
coming
never
in.
I don't know. Don't know pteropods or pseudo
Okay.
thecostomes.
Can I come in more, pilot?
Or any of them
Hold on
well
one
enough
second.
to
There you go. You can come in, yeah.
say what it might be. This looks like it must be a regular anemone rather than a cereanthid burrowing anemone, because I do not see an inner ring of tentacles.
It looks very similar to one that we've been seeing in the eastern tropical Pacific deep ocean, abyssal deep ocean, but we haven't been able to get an ID on it other than anemone, actinarium.
And 4 of
All
its
right, let me know what you
tentacles
wanna do,
are
video.
very small.
Okay,
They could be, I
let's see.
suppose,
I'm going to come in more
regenerating
if I
or
can.
All right, go ahead.
Okay, that's full
Thank
zoom,
you, Bridge.
pilot.
Copy.
I'm not sure of the sequence, but there are a total of 12 tentacles, which is in keeping with its being in the hexacoralia with the other anemones and stony corals. They have those tentacles and interior partitions in multiples of 6. Tina suggests it might be an Edwardsiid anemone. Edwardsiidae.
It looks like it's closing.
Thank you, pilot.
There's just.
If I remember, Tina, maybe you can
Okay.
say something in the
I think I'm
chat
good,
room.
pilot.
Aren't
All right, let's come out.
Edwardsiids
Okay.
sort of juvenilized, that, you know, they look like little
Here you go, pilot.
juvenile
All right.
anemones?
Bringing
I
camera
don't know
up.
if that's true of all members of the family.
Okay, losing skids.
Copy.
And now we're back into what looks like the terrain we were traversing at the very beginning of this dive. This very sedimented area,
That last
not a
move
lot
is
of
finished.
activity in in terms of chemosynthetic activity
Copy.
or geological activity, and a lot of these mounds with associated burrows as well.
And our depth is
Pilot at
770
45,
meters,
40
so we're
degrees.
still moving up.
Okay, yeah, if you want.
So if you want another move, I think we can.
There's nothing on
And
site.
there are a number
So now
of
it's
these
clear, this
tunnel
direction.
burrows
All right.
with two
Bridge,
entrances,
Charlie,
or
now.
an entrance and an exit.
Yeah, can we get another move?
Let's
Same as the last.
come in right here.
30
videos.
meters
Okay,
at
coming in.
270 degrees, 0.2.
That's partial.
Is that anything? Not right.
I don't think so. I think it's a
Okay.
hole.
All right, let's come up.
Good copy,
Coming out.
Bridge. We're watching that wind down here. Does it seem like it's holding all right?
And fish right there. I think we've seen it before.
Yeah,
but let's
roger
just
that. We
come
got
in
our
partially.
gauges up
Okay,
down
coming.
here, and I see that it's crept
It's a little
up
bit low.
for a
Okay.
while there at 20 knots.
And this
Keep us
one
updated.
is a
Thanks, Bridge.
ophidiid, I guess.
All right. Trying to hold that.
Yeah,
Is that full zoom?
No, I have more.
Hold on 1 second. All right, let's try to come in.
Okay, coming in.
Thank you, sir.
And
All right, let's
Tina
come
said
out
that
partially.
little
Okay, coming
anemone
out.
we saw,
There you go.
which might
Hold
be
right
an
there.
Edwardsiid,
There you go.
that family, I asked whether they were, they look like baby
All right,
anemones.
let's come out.
The term
Already
is
coming out.
maybe, I'm not sure if it's correct in this case or not,
That's full wide
paedomorphic,
pilot.
meaning
Thank you.
that the adult retains the appearance of a juvenile.
Okay. And losing
And
skids.
she checked and said that the family
laces
is
on.
at the
Thank you.
at the base
Ooh,
of the
there's a
anemone
sea star pilot.
phylogenetic
Yep,
tree.
that's where I'm going.
Excellent.
just been joined by Les Watling from the University of Hawaii. He's a coral expert, deep sea coral expert.
Let me
And he said
actually
he's
try to land on
just
this
been
one
flicking
video.
through the dive. And he
Okay.
has to say that the baby chimera that we saw two of is easily the cutest fish he has ever seen. He loves the adults, but because they always look like they're smiling. So this looks like a Hymenaster,
Yep.
which is a slime star.
All right.
And these are very
Let's come in video.
cushiony
Okay, coming in.
Hold right
sea
there.
stars. They look
Hold
very
it.
Second.
robust and thick. Oh, actually,
There you
no.
go. Come in a little
I'm
bit
gonna
more.
change
Okay,
that.
coming
Yeah,
in.
because the
Yes.
edge is
Yeah.
the edge is now visible.
Take that back. Not a Hymenaster.
No, and you can see, and there's a scale worm on the left side there, a little purple guy.
So this is what we were saying earlier when we were saying that many echinoderms have this scale worm that sort of
Is that
follows them
wholesome?
around.
I believe I have a little bit left.
Actually, let's
But
try
the
to keep
reason
it there
we,
because I don't think
Yeah.
I can.
misidentified this initially is
Hold
because
this.
it's partly buried, and you can see at the along the lower rim those
All
very
right, let
large
me know when you're
blocky,
good video.
Okay.
skeletal ossicles.
Can I try and
which
come in?
Hymenaster does not.
Yeah, let's come in.
So I'm guessing that this, I
that's
don't
full
know
zoom.
if this is a goniasterid. It looks like it also has a bunch of
Let me know when
little-
you're ready to come
some
out. I
kind
think
of
there's gonna be
commensals, little
Okay.
pink
some
things
dust coming
Okay.
into
crawling
view.
around
Coming
on
out.
it. I don't see
All
them
right.
moving, but they appear to irregularly distributed to be part of the sea star.
It could be like myxostomatids. And so Andrew was saying that ophidiid we were looking at in the little pit was a monometopus, which Ken had also suggested earlier. Thank you, Andrea and Ken.
Oh, this isn't great. Let's get out of this.
So you're looking at, yeah, got it.
Can you unlatch me? Please? Yeah. There we go.
Do you want me to unlatch you too?
If you say no,
Yeah,
I'll be offended.
if you have me latched. I prefer not to be latched over here because it's really hard to hear the bridge sometimes. But I will always be listening.
Copy that. co-pilot. Do you want me to latch you?
Yeah. Yeah, I like to hear your voice.
Copy.
Okay, lined up back west. And off we go.
So we've just had a watch change. I'm not sure if you guys want to introduce yourselves.
We would love to do that.
Thank you.
Hey guys, on shore, the watch has changed. Sitting at pilot now is Jeffrey Lanning. Sitting at my navigator position is
Bobby Moore, navigator.
Sitting at co-pilot,
Sean Kennison.
clipping or at video shading.
Is Caitlin Bailey?
And we have Bob Knott clipping in the back.
Yep.
And we're all here with the Global Foundation for Ocean Exploration, bringing you the bottom of the ocean live.
So those are our video engineers and ROV pilots and engineers. And additionally, in the control van, we've got your science party. So I am one of the biology co-leads. My name is Diva Amon, and I am a research fellow at the Natural History Museum in London. I tend to work on chemosynthetic habitats as well as human impact on the deep ocean.
I do. I think it's a snail.
Oh, it's another snail. We've seen a few
I'm
of these.
on the faculty. There's Gaza again. Beautiful gastropod.
Okay, we've seen a bunch of those.
Okay, coming out.
Thanks.
Dorsal side of its foot. I'm on in the Department of Marine and Environmental Sciences at Nova Southeastern University in Fort Lauderdale. My area of specialization is the crinoids, sea lilies and feather stars.
Let's do a scan
I also
out.
work on deep coral communities.
Towards north.
And I am Alex Avila. I am a PhD student at Oregon State University in Fisheries. I'm also a Dr. Nancy Foster Scholar, which is
Nothing on
administered by the
sonar.
Office of National Marine Sanctuaries. And I am also an
appealing.
Oregon State Sea Grant Fellow, and I'm here to help out with data collection and
That
sample
move's
processing.
wrapping up. You guys want another?
Yes, please.
50?
Yeah.
Sounds good.
So in addition to all the voices you've just heard, we are joined by a number of scientists ashore via telepresence. So this is where we, on the
They're
ship,
dealing
are
with
able
a squall
to
up there.
send
The
back.
winds have come up and shifted
whoa, there's a
around the
big crab
stern,
over there.
so we might have to hold
We are
off on
able
our
to
move.
send
but
back all of our data
we'll wait to hear
as
from
well
them.
as this live stream.
It's a giant
bridge.
crab.
To shore, and
Should we go check
scientists
out the
ashore
giant crab?
can access it
Yes, please.
Sounds
within
great.
2
Then
or
let's put
3
in another
seconds
move, please.
of it being collected. which is
50
just
meters
incredible.
at 270
Joining
decimal
us
2.
today, we have Andrea
Near free
Quattrini,
will to turn the lasers
a
on
fish
and off
and
as you
coral
feel
expert
it.
who's
Good
worked
copy.
with
Thank you,
Mexico
Bridge.
quite a
No need
lot.
for requests.
Andrew Shula, Asako Matsumoto from Japan, a Nigerian expert.
Humongo
Carolyn
bungo
Rupel, a
crab.
geologist at USGS.
The body is 10
Ken
centimeters wide.
Sulak, an ichthyologist who's had a lot of expertise working in a lot of experience, working in the Caribbean and the Gulf of Mexico.
Right, I'm gonna ease
Les
off
Watling
here on my
from the
forward
University
way.
of Hawaii, again a
we're
Nigerian
good on the
expert.
lasers.
Mark Benfield, who
Wanna
has
come in
worked in the
partial
Gulf of
on it?
Mexico extensively.
Okay, coming in
Megan
partial.
McCullough, who tends to work on bryozoans. Bob Carney, again,
Good
another
copy.
person
Thank you,
who has
Bridge.
had
We
a
found
lot of
one less,
experience working in the
sediment
Gulf of
covered.
Mexico. Scott France, a coral expert
get a
from the
little
University of
closer.
Louisiana Lafayette. And
ease
Tina
off.
Molotova,
Move
who
is
is
in.
a coral expert joining us
Okay.
from Russia at the Shirshov Institute of Oceanology.
There's
so
a
thank
Tina
you
for
everyone
Riot,
for
too.
sticking
Yeah.
with us throughout this dive. We have about an hour and 15 minutes left.
Okay.
We are at a
Okay.
depth
Just
of
pirouette around the sky a little bit.
768
We've got
Sounds good.
meters, roughly, and are approaching
Hopefully I don't kick up
waypoint
any
2,
dirt.
which
I'm
will
a
be
little
on
low.
the
Can
high
I come out
of
just a
this
little bit to get his legs?
we
I'll
were
tilt down
hoping
for that
to
if
see
you
Oh, okay.
some
Oh, that's a lot
hard
better.
substrate
Thank
there,
you.
but
Yep.
so far we haven't come across any. Once we get to waypoint 2, we'll begin moving south along that ridge feature.
And this is a lithodid king crab, I think in the genus Neolithodes, and you can see it's not a true crab because following its claws there, it only has 3 walking legs instead of the 4 pairs of true crabs. This one, the 4th pair is much smaller and they're tucked up underneath the carapace. And so this places
Pick
it
up a
in
little.
the
Maybe
mo-
we can go for the moving parts
with
there.
the
Come in a little tighter in
hermit
its
crabs
face.
Okay.
and the squat
Coming in?
lobsters.
Yep.
And there's a wonderful view of all of the mouth parts. Between the claws, the 2 little legs that are hanging down and opening wide there as the animal puts something up in its
That's beautiful,
mouth.
Jeff.
Those are the third maxillipeds, and the other mouth parts are sort of stacked up above it towards the mouth. There'll be 2 more pairs of maxillipeds. Those are actually thoracic appendages modified for food handling. Then there'll be the head appendages, which are a pair of maxillae.
I was just about to say that I have more zoom.
One of them going back and forth there.
Okay, coming out.
Thanks.
And those 2 little structures going up and down on either side are sort of boat-shaped extensions of one of the mouth parts that generate the flow through the gill chamber under the carapace. Because these guys have no cilia, the water has to be pulled through by the movement of this little boat-shaped extension off the side of one of the mouth parts.
So Les Watling was suggesting that crab looks very clean compared to a lot of the other crabs that we've been seeing during this dive. None of the species so far, but many other crabs, and perhaps this one may have just shed its exoskeleton or molted, and that's why it looks so fresh and so clean.
There's something moving straight ahead, Pilot. See that?
Yep. Another crab.
There's some discussion in the chat room how an animal this large does not sink into the sediment.
Much smaller this time.
And one of the possibilities is that
come on in.
Yeah, I think it's a pagurrid.
Ends of its legs are covered with bristles that give it more of a snowshoe effect.
I suppose we've got
There's
plenty
a
of
little
this then.
hermit crab.
Actually, it looks a little bit different from the other ones, but I don't know if we have this or not.
Come in partial.
Okay,
time.
Coming in.
Thanks.
And it has very bushy looking bristles, sort of sediment covered on its claws there. They're brown, and so the walking legs are white and red, and you can see its, I guess those are the second antennae. I have to double check. All crustaceans, unless they're extremely modified as parasites, for example, have 2 pairs of antennae.
When you're ready, I'm ready.
Okay,
which have
coming
one
out.
pair, and of course arachnids have none.
Off we go.
And I'll have to
that's
double
full
check whether those 2 little short ones were I think those were the, I can never remember if they were the first antennae or not. And we just zipped by. There was somebody at the entrance to one of those tunnel burrows.
Yeah, and he ran in.
Went back inside, and we missed it. It's a crustacean of some sort.
Could we get a look at, that pile of stuff?
Yep, headed for it.
Oh, and before that, there's this little animal. Oh no, it's a jelly just above the seafloor.
Oh, I see.
There,
yeah.
It's kind of to stop and not disturb it.
Bob Carney suggested that hermit might be Parapagurus pilosomanus, which means hairy hand. And there's one of those little flapping gastropods again, above just one off screen. And here's another Medusa.
Settle here on it. Okay, come on in.
Coming in.
Pinky color.
Oh, no.
And there it goes.
He heard me say zoom.
Probably very disturbed by a combination of our lights and the bow wave of D2.
Okay, you can come in.
Can I come in?
Relaxed a little.
Excellent.
Marsh is listening.
Oh wow, beautiful.
That's gorgeous. So you can see those. I believe those.
Lost
reddish ovals in the interior may have been the gonads.
Oh, we got a crab up there. Lobster.
Pilot, co-pilot, we're nearing the area.
Pile.
We're nearing the area where we're looking for a ridge. So keep your eyes out for anything we can follow south.
One of the trail makers is one of the gastropods.
Want to go in partial on this
So
pile?
this may be a
coming in.
A molt?
I don't know what it is.
It does look like a molt, yeah.
Oh,
yeah,
it
it
does.
eaten by Phimorhynchus. gastropods, which are one of the main scavenging predatory gastropods found at cold seeps and so
So
on.
is that likely a molt or a dead?
Or a dead one, maybe.
A dead one.
And this looks like a Chaseon quinquedin.
Yes,
Predation event. I guess we like them.
Yeah.
So that's one of the red crabs, which are commercially fished in this area.
That crab on the left doesn't care at all.
Be interesting if anybody knows whether there's enough nutrition on a molted
I have more
exoskeleton
Zoom pilot.
to warrant
Yeah, sure.
such
Coming in.
attention.
I think there would be. I mean, a lot of animals actually eat their own molts.
Okay.
And that's
It
to
looks
try
like there's
and
a fish
replenish
hiding on the left.
the nutrients they may have just
We
lost.
saw one
So
of those
why would
earlier
it not
today.
be
That kind of
a
fish,
good source of food
sedentary,
for
just hangs
some
out.
other animal?
And there are lots of little gooseneck
but
barnacles
looks like I need to
still alive
catch up
on
once
this
we get
molt.
this
Aren't
shot.
most
We're
molds like a
happy.
lot lighter in color then?
No.
No.
I think it depends what color the animal was when it molted. So the molt will change color throughout the animal's life. Usually just before many crabs
It's a barnacle on it.
or lobsters molt, it'll actually darken. So this blackened stuff
Alright.
you'll see.
I should play the
see
catch-up
here,
game.
It'll,
Okay.
become a little more black.
Coming out.
And
Next
there's
video.
a zoarcid puddling under the legs on
that's
the
full white
left-hand
pilot.
side.
Thank you.
Perhaps those snails also might have been feeding on the gooseneck barnacles.
Okay, looking for some sort of ridge to follow.
Look at all of those track marks.
Yeah.
So that's essentially like, a big parcel of food, and animals have come from far and wide to feed on that shell or body. And yeah, you can really see how far they've come.
And there was a brittle star there, and off to one side, and a couple of squat lobsters.
Watch lead nav.
Go ahead, Nav.
Yeah, I have a question relating to these tracks. Do we think that is intentional? I mean, are they following some sort of scent or something else towards the crab molting? Or is it just luck of the crawl?
Nope, absolutely following a scent. So just like how we are drawn to the dinner table by the smell of freshly cooked brownies or whatever it may be, these animals will also be drawn by a slick, if you will, of chemicals in the water being emitted from that carcass, and they will be able to follow that trail down, which is usually up current, obviously, to get to that carcass. Additionally, in the case of some, not necessarily this, which looks like it may have been dead for a while or scavenged, but if, for instance, a fish carcass came to the sea floor, there was some animal that was struggling and dying, those vibrations and that movement that it's giving off would be then carried through the water column, and many animals do, in addition to their chemosensory organs, would be able to pick up those mechanosensory clues. So being able to smell as well as feel potential food opportunities in the distance.
Great, thank you.
Great question. Thanks, Bobby.
And you'll also, of course, see the trails.
Pilots, that ship move is wrapping
regardless
up.
as these animals cruise the
You're kind of
sediment
right
surface,
there in the
like
vicinity
that
of the
one right
ridge that
there.
they're looking for on high
And there's
pack.
another
Yeah, I
trickle
think we're
carrion
on
crab.
a little high here, but it doesn't seem to go uphill in either the north or south.
Watch leads, Nav.
Go ahead.
Yes, a position question now.
Might have something.
On high pack, we're at waypoint 2,
but it's not
our
prominent.
target, but we're not seeing a very definitive ridge, as you can see. What's your pleasure? Should we continue uphill? Should we turn south?
How much further does high pack indicate this ridge goes on to the high, roughly?
So there's a zoomed out version.
Yeah.
If we go right here to center, that's 40 meters.
Yeah, so if we could just continue to there, just to make sure we're missing any hard substrate,
Thank you,
and then
Bridge.
we'll work our way south from here.
You got it. We'll continue on.
We got a couple of returns here, Bobby, that might be, I don't know, might be something. You see it in the D2 sonar?
You're looking, what?
Can you go ahead and check them out?
Alright, I'm gonna push out that way then.
Oh, that's south, yeah.
It's towards the south.
Might be something. I can turn with you. And we can reach out a good bit.
Okay.
So, yeah, to center of this highlight on high pack, which
Here's some, yeah. There must be
is
some.
30 meters, so maybe just go around at tether length.
Okay.
And looks like we've got something.
Woo,
So this is
hard
towards the
substrate.
south.
along the ridge, or along the what Pipac calls
Looks
the
very
direction of the
similar
ridge.
to what we saw earlier with the elongated ridge of outcrop, broken, and running along the upslope there.
Okay. We can do that.
So it looks like we
These
may have
little
a
downhills,
first
we're just
coral
getting ourselves oriented
of
here.
this dive.
Yes.
Oh, there's a coral coming into view. Hold on.
And more in the distance once
Backing
we
up.
check this out.
There's a
There's also
whitish,
one-
bluish one right below me here.
Yeah.
Oh yeah, a couple
Tilting
corals.
down.
Brilliant.
Yep, that looks.
There you
oh,
go.
yes,
beautiful.
indeed.
That's the one.
So again, this, as Chuck was saying, this feature looks really
Yeah,
similar to what we
we're
were
too
seeing
far
before.
down for lasers.
It's a very long
right there.
feature, elongated feature.
The move is complete.
Which is
Let me
probably
see.
carbonate,
Alright.
a carbonate
Sorry,
outcrop of some
sometimes I
kind.
get
I'm not
colorblind.
sure whether it would be orthogenic
Sometimes.
or otherwise. Probably orthogenic, given the
Alright, lasers are coming
the
off.
sort of
Thanks.
vugginess of
You
it.
can go past the skids at least
There's
here.
also many of these chemosynthetic clamshells on both sides of it, but this one, which is nice and different to the one we were looking at before, has corals growing on it. The one we visited before did not. It had more brachiopods, cesta bivalves,
Can come in a
encrusting
little more.
sponges,
But
and
I
so on.
rotated
So this looks like a
25
different
degrees
community.
to start or
And
to
this
port to keep
also,
in
it
view.
looks
the ship?
like
The ship is stopped.
the
Okay.
whole thing is like a
Roger,
tilted
thanks.
outcrop, like it was originally laid down flat and then tilted
I
up.
have more
but
to Zoom Pilot.
I don't know enough about
Okay.
this sort
Yeah,
of
you
thing.
can have it if you want.
So
Okay,
any of
coming
our
in.
coral folks across ashore, Tina suggested Acanthogorgia.
Yeah, so this looks kind of similar to the one we collected. Do you
Okay,
remember? We
yes,
collected one of
yes.
these on an earlier dive. There was this light, beautiful purple, and I'm pretty sure that was an Acanthogorgia
Sneaking
as
in
well.
on it still, trying to slow
Yeah,
down.
so Scott just said, I think we collected one like this earlier in the cruise. This just looks like a bigger colony than the one we had sampled. So pretty. So this is a deep sea octocoral. We can tell that because each polyp has 8 pinnules. 8 tentacles, and then on each tentacle there are a number of pinnules. This is likely from the family Acanthogorgidae and the genus Acanthogorgia. So now there's some debate happening in the chat room. Scott suggested this may be one we collected earlier in the cruise, as did I. And Tina and Asako were saying, no, they think this is different.
Maybe we should collect this too.
So folks, is this something that you would potentially want to collect a piece of?
There's a little shrimp in there, perhaps Bathypaleomanella.
Oops. Ooh, there's a little hydrite or something on the end there. Sorry, just bouncing around
It's okay.
here. I don't have much of a foothold. I'm just leaning
No, now
against the
Tina's
rock
saying
in
it
the middle
has
of
to
my
be a
skid.
Plexauridae because the calices are visible.
And Scott is checking out the previous samples. So hang tight, group, and we'll see. Again, this is just remarkable. We've got Scott in Louisiana. We've got Asako in Japan and Tina in Russia discussing the possible
Is there a
identity
section that you like in
of
particular that I'm going to get?
There was that shrimp
an
to the
octocoral
right
that we are
that
observing
they commented on.
in 760 meters on the floor of the Gulf of Mexico. It's just wonderful.
There he is. It's being shy.
I mean, just think about that.
And
That's just amazing.
it is.
And regardless of their decision, it still points out the fact that in so many cases, you really need the specimen to look at the microscopic components of their anatomy and, of course, to extract DNA to compare them with other specimens and reconstruct their
Can I
evolutionary
come partial
history.
to get more of a
And
wider
not just
view?
that, but see how they connect with
great.
other individuals, and that can
Now
give
I
us an
have
idea
a-
of which
there we go.
populations reproduce with which ones in this area, which is pretty important from a management point of view.
So we've got Asako and Les Watling, who's out in Hawaii. That's another point on the globe saying that it's not an acanthogorgiid.
Okay, so it's definitely a plexaurid. Some folks are saying it looks different. Scott, you're looking at the previous sample pictures, so this kind of comes down to you being able to tell us whether this looks very similar to the one we collected before.
Andrea says we should collect.
I'm going to come back in, pilot.
Okay.
Or maybe not.
We've had several.
Yeah, why don't you come
Let's
out? I'm going to
collect.
set up
so
for a collection. We can zoom
yeah,
back
pilot,
in
we
once
wanna
I get
collect
a better
some of
footing
this,
here.
please.
Okay, sounds good. That's a full white pilot.
Thank you.
Nav,
So now Scott
for
says
samples,
this
do we
looks
just
like
have
the
a
antherthelid
rock?
that we collected on dive
I've
9.
got the rock in the port inner.
Yeah, it's a rock listed as port inner instead of the rock box. It looks like they put it in the bio.
All right, Pat, so you got port outer, starboard inner, starboard outer.
So we will collect. And I'm guessing
come in
that
partial on it, Bill?
Okay,
the
coming in
orange
partial.
ones upper left, the orange octacals are probably Swiftia.
Is that good, pilot?
Yeah, that's good. Watch lead. Which section are you hoping to get from?
It doesn't really matter. It's not like a bamboo coral where they'll say we need to get nodes, for instance. So,
Okay. So see these two branches sort of sticking out, or one of those okay?
yeah, so just one of
Sticking
those would
towards
be
the right.
fine. Thank you.
Awesome.
All right, you're ramped up.
Thank you.
Yeah, so Tina's asking us the
All
same
right,
thing.
can
What
we
is the
come
red
wide
one
just
off
so I
to
can
the
line
left?
it up?
So we
Okay,
will go
coming
there
wide.
after
We'll want to
check
go right
out
back
what
to that when I go to do
other
the grab.
colony is.
Sounds
There
good.
were also a couple colonies
you.
ahead on
What
another
camera
large
would you
red
like?
one, and some
Up
of them
on
look
my
sort of
screen.
pinky.
The Zeus is
And
going to
there may be
get me there.
brachiopods under the edge of the rock at the top of your field of view. There is at least one cup coral down left of lower center attached to the rock.
Yeah, how brittle is this going to be, I wonder? Seems not too bad. Grip force more than 2. There we go. Okay, you can come in partial again.
Okay, coming in.
Just so I can watch where my fingers are going. Make sure I don't take more than I want.
Do you need more, pilot?
Nope, that's perfect. Thank you. They are going to go below that stem. Don't want all that. Maybe I should come in from the side. Don't want to collect so much of it. Really likes to cling to the claws. I thought it would just snick right through. Jeez. Like Velcro. Maybe the lower branch is the way to go. Is that going to be enough, or do I need more of it?
Nope, that will be just fine. Thank you.
Okay. ship rolls. No, don't cut here.
No, I'm sorry. I was just making noises with the rolling of the ship. Ignore me. Sorry, I'll mute myself.
That's okay. I just want to make sure that was not a cut somewhere different noise. That's it.
Alright, pilot, do you know what box you'd like?
It doesn't matter. I'll go starboard.
Starboard?
Just stuff on the port side.
Copy.
Keep things separated if we can.
Starboard.
That's full white pilot.
Then ready to extend?
Yep.
Coming out. It's fully out.
Okay. And deposit the sample into the box.
It's in.
It's in. Copy. Jeez.
Alright, retracting. Good job.
Wonderful.
Yeah, thank you very much, pilot. And in future, you know I'm not subtle. I would just be like, don't cut that. Let's cut something else.
Yeah, I'm just making
I
sure.
wouldn't make subtle noises.
Just making
so
sure.
that was collected in 761 meters.
Okay.
And we'll see what it is eventually. We could take a look at the orange ones once you gentlemen get settled.
Yep, of course.
And in serious view, you can see how really narrow this outcrop is, and now you can see as well in...
Would you like to go for that large orange colony or the small one close by?
They're probably the same. So if we could do this one with the brittle star, and then the larger orange one would be great.
Okay.
But I'm going to keep rotating. Keep a better view, yeah.
Thanks. Sounds great. There's a little bit of current here now. Just a smidge.
So I'm guessing this
okay,
pale
you can start in.
Okay, coming in.
Seafan octocarl may be a paramuracea.
Partial.
Thank
with
you.
the Ophiochreus brittle star in it, but there are some thick structures there that I
light
don't
it up for you
recognize
here.
offhand.
Thank you.
Oh, that's interesting.
And
Okay.
a
Oh, there's some
a
eggs.
cesta just
Egg
visible
sacs on
below
there, I
it.
think.
On the
Is that
side
what
of
that
the
is?
I don't know what those are.
Maybe.
Well, there's an open one and then some growths
What
or an
are
egg sac
those?
there. Come on in.
Okay.
Scott agrees.
Perfect.
Paramuracea.
Thanks.
Okay. I have a little bit
And
more,
those
but
look like some kind of
this is a nice shot.
swelling, maybe a
Okay.
parasite of some
Yeah.
sort.
No need to take it
Because
if
it
you
looks
don't want it.
like there are still polyps growing out of it if reduced. Or is that an egg case of some sort?
Yeah, see, I was thinking egg case, and then
Okay.
tissue had grown over.
Okay. And
But
then,
I am not a coral person, so...
and this is a different species. I don't know if this
No.
is Ophiochreus.
So then Scott's saying no, he thinks it's parasitized.
Okay. And this
Sorry, getting a
brittle
little
star
close.
certainly looks
You're okay.
different than the ones we've seen. Instead of that smooth tissue covering its arms, it's got little papillae or spines all over it, so I don't know if it's in the same genus or not. Scott suggests it might be an Ascothoracicon, which is a highly modified kind of barnacle relative. And they do parasitize. There's a third one up there. They do parasitize octocarls.
Hmm?
But then at the top there looks like the
You're muted.
shell of one.
Having trouble
Or that
hearing
could be
you.
the
Sorry.
shell of a Dumbo octopus, as we've
I
seen
was just
on
saying it's one of those
several,
curly
I think we
Q
saw one
worms.
on this expedition
Those
as
are always
well
cool
as, oh, and then there's
to
also
me.
Aplacophorans.
Oh yeah,
one.
that curly
two
Q
of them.
worms, those pale
Three of
yellow.
them.
Yeah, the pale yellow coiled worms
Four of
around the
them.
branches.
So there's a lot going on here. Oh, and then, so I disagree. I think these are definitely shark egg cases that have had tissue grown over them. Because this coiling here looks like what they attach their eggs to.
Okay.
Eggs with.
The egg case, yes.
Yeah.
Okay.
But then this egg case right here looks more like circular. So that could be like a dump rock.
Well, it could be the end of
maybe.
it broken off.
But
That's
then
a
Scott's
good call.
saying he's never seen a plexaurid overgrow things that were deposited on the colony. But then those squiggly coily things do look like what sharks used to attach their egg cases to.
Can we get a close-up of the one below there? Yes.
And then it has that ribbing on the
I do have more zoom
side of it.
Yeah,
the edges of
I don't
it.
know if I can
like
keep stable. You can try it.
I can try.
Rank egg case, yes.
That's full zoom.
And clearly they've overgrown it there. Those aren't zoanthids, those are octocoral polyps. And that's a different snake star than I've seen. Especially this one I like as an old egg case.
Okay.
With the ribs along the edge, the
bouncing around a little here.
portions and shape.
We're getting closer to it, I think. Yeah.
Definitely moving around. Go.
Really
I think I've
interesting.
set
Yeah,
my
see,
front
look what I
skid down
mean.
on the rock barely, so should be a little bit more stable.
Yep.
Yeah, if you just Google mermaid's purse, you do get some images which do appear to have those sort of coiling structures they attach with, as well as structures that look very similar to this. I mean, Scott, could this be the first time that we're seeing something like this, a plexaurid which grows over structures that may have been attached? And how quickly would this have to grow over for...
It's between two branches. You see that?
Oh, yeah.
You see that? You can see two branch ends
I
sticking
do
out.
see what you mean.
Maybe the egg grew over the coral. Don't know.
Okay, well, we stumped everybody. Which is a good sign. Okay, well, if we could move on to our next colony then, please.
Yep, of course. When you're ready, video.
Okay, I'm ready. Coming up.
Thank you.
That was cool.
That's full wide,
really
pilot.
cool.
Thank you.
Wonder what kind of sharks deposit their eggs down here, if it is that.
Or do skates do that?
Oh yeah, because we have seen a couple. Yes, they do, and we have seen a couple of those.
Something attached to this coral too. A couple things. I think I'm gonna try to lean against this rock on my right.
Ooh, this one definitely has
Hopefully get a better
some
stable shot.
newer looking
lasers.
ones attached.
Thanks, co-pilot.
This could solve the
That
mystery
looks pretty good. Coming off.
of the day.
Okay, video. Can start in.
Okay, coming in.
I think I'll be a little bit more stable this time.
Okay, this is a
toehold.
partial full specimen view.
Thanks.
Suspense is killing us.
Looks like there's a egg on the left and a egg up on top.
And I wonder what those little tiny white dots are on
gonna
the
come
rock
in.
surface
Okay.
to the
We can. Oh, what are you?
to the
Oh,
right.
that's not
I'll
those.
check that out later.
That looks like
And here they're overgrown too.
it's torn open.
Look at that.
And it's
But definitely looks like it's broken open,
Yes.
and something
Yes.
may have
That's
left
full zoom, pilot.
And there's one
Thank you.
above this. If we could pan upward a bit.
Yeah, of
Which
course.
is clean.
I'm going to give this a couple seconds for video's sake,
Yeah,
and then I'll
of course.
pan up to the
This
green
is
one.
Really interesting.
It's cool. It looks like it has a face. So. Okay, we can go up now.
Okay, I'm going to tilt
Tilt up.
Yeah. And turn over this.
There it
way.
is.
Ooh.
Looks like the husk of a tamale.
So that is that
To be
is
honest.
definitely
An
some
elasma
kind of egg
blank
case.
yes.
Yes. So
Interesting.
I mean, I think this is a first for a lot of these coral taxonomists, coral biologists joining us via telepresence.
And Scott points out that this indicates that these octocorals that have a protein axis that grow upright with an internal support still have the genetic, I guess, capability of growing mat-like, like the related groups, the stoloniferans, which only grow mat-like.
Oh,
and
that is so true.
Clavulariids. Good, really interesting point, Scott.
Yeah, and that's kind of mind-blowing.
50 minutes.
This is amazing.
There's a little amphipod in the center there on one of the branches.
So is this a Swiftia, folks?
Asako said that she thought
I was spotted
so
this morning.
earlier.
Okay.
I'm actually supposed to be sitting nav.
Yeah, a couple little amphipods here, but wow, this is amazing. Some kind of elasmobranch egg cases, sharks or rays, down at 760 meters, which is not uncommon, but attached to 2 different types of corals. Some look intact but overgrown by the coral tissue, and some hatched and overgrown, and then some hatched and not overgrown. So I wonder if they are not hatched, whether they can hatch if they're overgrown.
Well, I would, my guess is that if they are viable, that they would hatch long before the octocoral had the opportunity to overgrow them.
So then this begs all that questions about growth rates and...
well, sure.
Yeah, like coral growth rate versus growth rate of the embryo.
Don't know. Andrea Quattrini has mentioned that these are probably cat shark egg cases.
We'll Google that.
So here you have one that broke open and released its offspring and is not overgrown. So my guess, I would bet that collect my thoughts, Chuck, that if it is overgrown, it takes place
I wonder if there's anything
at a much
inside
slower
the egg
rate
case.
than the
yeah.
gestation period of the cat shark. And if it's an intact one, an intact egg case, perhaps the egg case was not viable.
They're holding real well.
And
copy.
I mean,
Tina suggests that
I mean, it might be
we talked about
2 meters.
We talked
They're
about the
holding
idea
strong.
of these, this octocoral being able to grow mat-like and encrust over these egg cases. She says that
they've actually been doing great for the last hour or so. You could read all their moves based on where they're drilling holes.
Can you repeat?
Underlying the mat of
coming out.
encrusted polyps.
That's
really
why
interesting discussion here. So also, you know, for any of you
Oh,
who...
the paraphila now stuck in the coral. If we could just get a quick zoom.
Look at that. Lost its way.
Okay, coming in.
Yep, it's caught in there.
So many of these around, and so many of them looking like they're generally having a really bad time.
But what you're listening to and seeing here in our discussion about
there's another egg case on the left.
These
I think.
octocorals, their identity, the way they grow, these are all among the first steps that professionals in this sort of discipline take and must take before coming to conclusions
deck and then
about
winch, so I really
particularly interested in filming it. So
whose identity is not well known.
Makes it kind of sad.
If
Okay,
known at all.
Let's go.
Sometimes there's an aha moment when you are looking at an organism, let's say within your discipline.
This is looking pretty good, like a ridge. So maybe we just want to follow this south.
It looks strange, and it looks strange, and then you have that aha moment where you see a character that you read about previously,
195.
and you say, aha,
might as well just keep going from sonar target to sonar target until we don't find any more.
Back in 1915, I remember
Copy that.
image
Let me give you a move,
in his
co-pilot.
paper, and there
I'm
it is
looking like
staring you in the face.
195.
Okay.
So plus or minus in that general area.
What do you want for 195?
Outcrops here with
Do 30 meters. I think it's fine.
Okay.
pale purple octocorals left and orange octocorals center, right? Which the ones in the center look more like Swiftia, and not sure about the ones to the left. If we could take a zoom on those.
Bridge, ROV Nav.
They may be the same thing as the one we saw.
Yes, can we get a
but they
move,
look
please,
more
of
monoplanar.
30 meters, bearing
They're standing up.
195,
They're
0.2
not tucked
knots.
down on the side of a ledge, like the previous
Okay,
one that
coming
we
in
collected
partial.
was. But I'm not sure.
Good copy, thank you, Bridge. Go ahead, Watchley. I can't hear you.
Sorry. Are we heading south right now?
I still can't hear you. But yes, we're following the south unless you want to continue west. That was our plan up here. Great. Thank you, Bridge.
So can we
Okay.
get a zoom
Coming
on
in.
these when you are ready? Thank you. So, group ashore, is this the same one as the one we collected a little while ago?
Or zoom? I think
It's the same color at any
so.
rate.
That's full zoom.
I'm not sure if it's the same or not.
Can I come out a little, or?
Yeah, Watchley, I think I'm hearing you over the air here, not over the microphone.
And Tina says it looks the same. So we've got a specimen of this, and we will find out what it is.
Okay, can we, how, we're leaving the bottom at 4, right? Now?
Working on your update right now.
Okay,
We
coming
have
in.
about 40 minutes left on bottom.
Thank you. No, I think we're okay actually.
This one?
Looks, did they say it looks like the same?
Yes.
Yeah.
Yes.
Yeah, so if we
We've
could,
got agreement from Louisiana, Japan, and Russia. So,
Okay, coming out.
you know,
it's pretty good.
that's pretty good.
So if we could continue moving along.
That's full
and,
wide.
we have about 40 minutes, a little less than 40 minutes left on the bottom here today.
Can I lose goods
at
pilot?
our site known as
There we go.
Penchant Basin. And finally, we've gotten to these areas of rock outcrops that we were hoping for, so that we've been able to. We can always get another zoom.
I'm not quite sure.
Where we
yeah.
were,
Check out
yeah.
that rat tail. And for that, the pronunciation of our site, Penchant, it's spelled Penchant, right?
Yeah.
So, what's the derivation? I mean, it sounds
right.
French.
I think it's
There you go.
French Creole.
French
from
Creole.
New
okay.
Orleans, like Louisiana, New Orleans,
Okay.
Stilio.
Come down a little bit, give you a little more. And I'm moving towards you too, so.
Okay,
And
coming
this
in.
one has a parasite in its dorsal fin. This is another macrurid. I'm not sure which one. And if
Okay.
Andrea or someone else ashore who knows about these
I
things,
think that's full zoom.
are those, what group of copepods, if that's what they are the parasites? There's one on the pelvic fin there and one up on the dorsal fin. I know there's a number of parasitic groups of copepods. Penellids, Dougal Lindsay says.
Dougal.
That's the name of the group to which these copepod parasites belong. Copepods, of course, are among the most important organisms in the ocean. The free-living planktonic forms are sometimes called the insects of the sea, and they are major components of ocean food webs throughout the world. Hundreds and hundreds of species. And the rat tail here is in the family Macruridae, and it is one of the most abundant and diverse families of fishes on this deep sea floor.
Yeah. He's getting closer to us.
Dougal says usually these
coming out.
Copepod parasites usually have
that's
a
full
pair
wide.
of long egg strings trailing behind, which we do not see here.
Our video is going
There's
to
another,
switch out.
Acesta, big file clam bivalve.
Yep.
Good afternoon, pilots.
And another one of those purple
Video.
octocorals that we've
How's it going?
seen, or two of them.
Thank you, Bridge.
We can zoom in on the one on the left. Tough to say if it's the same or not. Oh, that may be different. Is that a black
just
coral?
a quick zoom. It looks a bit different.
Is that a black coral?
And then also that big shell underneath.
And that's Acesta, I think. There are a couple of species, I think
I was
Bob
gonna say,
Carney
this one
said.
just looks not quite like what we were looking at before.
Dougal says that the first 5 or 6 segments of that parasitic copepod's body is usually buried or burrowed into the flesh of its host. Yeah, this is a black coral.
Ship move complete.
The bottle brush.
Copy
Look
that.
at the amount of those curly things you see are the cords that those eggs would be attached to, those elasmobranch eggs. And so these corals in this area are clearly very heavily used by elasmobranchs to lay their eggs in. This is the first time we've seen this during this expedition, and there are a lot of examples of these, the potential use of these by elasmobranchs in this area.
We've got a bit of discussion about the parasites. Andrea Quattrini says they're isopod crustaceans, so I'll let them hash it out. I thought penellids were much longer than that. I know the one penella that parasitizes large fish like the oceanic sunfish is a very long, slender one, but that's the only one I'm
right
familiar
over your pilot.
So
It's
these are
about
black corals.
9-0.
Tina, if you're there.
Move has been done for 2 minutes.
Copies. I'm just finishing up.
So we have some consensus that those parasites were probably isopods. And there's a little squat lobster with its claw sticking out.
And Tina, are these little balls we can see in the black coral, are those eggs? Every now and again you'll see some little orangey
white
balls.
Yeah.
But this is just amazing, the use of these corals. So deep sea coral habitats are a habitat forming
Ready for full screen?
group. They create 3D complex structures which are then utilized by a range of animals. So we can see here, for instance, there are many squat lobsters hanging out in this black coral, as well as we see ophiuroids, shrimp, amphipods, aplacophorans, lots of different animals find a home
that a shrimp
within
on the
these
right?
structures. But here we're also seeing a different use. These elasmobranchs are using these corals in this area to
Pink
lay
thing
their
on the
eggs.
upper right branch. I think it is
So
a
Tina
shrimp.
suggests this is Cibopathes macrospina.
Sure.
Thank you, Tina.
Very fascinating.
And there's a nice little shrimp in there. And this one has eggs. You can see those bright maroon colored eggs that are held in a cluster, so that shrimp is actually upside down with its head facing to the right and tail to the left. And so that's a Caridian shrimp. The two biggest groups of true shrimps, the Caridians, carry their eggs under their abdomen, and the Penaids, which are most of the, or many of the commercial species, do not. They release their eggs into the water.
Yeah, Pat, I think I'm settled. I'm not moving anymore.
And again, lots of these vesicomyid clamshells, dead. One of these Acesta bivalves, and also some
Brachiopods up underneath.
Yeah, so it just threw me a bit because this one's closed and looked, I guess, a little less oval than the other one. But yes, definitely a Acesta, which is a bivalve that has very bright orange insides and is usually found growing on other outcrops for instance, on canyon walls in the Atlantic and so on. Very, very common. That's great, Pilot. Thank you.
And lots of snail trails.
You also have a large boulder out to your left to go explore.
Oh.
What's that? That's some kind of octocoral. Ropey.
That should be enough, but we're not sure what this is. Hopefully on shore we'll be able to... I think this might be a black coral skeleton with overgrowing.
By stoloniferan. That could be.
Folks ashore, thoughts? Okay, that's great, Pilot. Thank you.
Could we get lasers on this before you zoom out?
Video's clear.
As well as, I think Tina was actually asking if we could get a quick look at that black coral we were just looking at with the lasers so she could gauge the size of it.
Right. So that colony is maybe 20 centimeters across.
So Les is saying that looks like an Acanthogorgia.
Full wide.
And so there the lasers look like they're on the colony there. See, as we move across. So that is probably 1.5 foot across. Maybe a little less than half a meter across. And the consensus was that little pale purple one was a... yeah,
I don't see anything.
It's a small one in sonar, straight ahead.
Anything.
I was misled by your port manip
Another
cam.
one of these
Has like
paraphilas.
fingerprints or something up there. I thought those
This
were
is probably
sponges
the
on the top of a
20th one we
boulder.
have seen on the
At
dive.
least.
They just seem
Those
to
on
be
the monitor?
everywhere.
Huh.
But at least that one wasn't trying to plow itself into the seafloor, as we've seen very often. Oh, and here we've got an
large
Acanthocaris.
lobster-looking
there
thing.
we go. Sika.
Blind lobster.
Floating. Yeah.
Let's do the floating first. And then...
I'm interested.
Can't quite
What
tell
is
what that
that?
is. Schmutz.
You got it.
No, it looks like a siphonophore.
Oh yeah, it's a siphonophore upside down.
What is going on?
It's just pointed downward. The swimming bells with the gas gland at the tip.
So luckily we've been joined by Dougal Lindsay, and he's a midwater expert based at Jamstack in Japan. So Dougal, you've come in at a good time.
And he's suggesting apolemia.
Wow, so beautiful.
So again, these bizarre organisms are colonies that behave like a single organism, in that they have directional swimming, for example.
On the clump.
But each one of the thing starts out as a little larva, and as it buds, produces the colony, it will create a gas gland up at the tip. It will bud off, or it will produce structures that are modified medusae, or quote-unquote jellyfish, that remain attached and act as the swimming bells, which are just out of sight now. And then there's this long rope or strand
go up to the
of
swimming bells.
Additional. There you can see some of the swimming bells. And the budding zone at the base of the swimming bells produces this long strand of what are called zooids, which may consist of the forms that have the stinging tentacle, forms that have a mouth, forms that are reproductive. So you have the stinging tentacle form is called a dactylozoid. The feeding kind is called a gastrozoid, very appropriate. And the reproductive kind is called a gonozoid. And then there are also modified medusae that act as sort of protective structures that are called bracts. And some of these species can be extremely long.
My video is clear.
Of course, they are predatory. They use their stinging apparatus to capture small prey. That
Absolutely
was
beautiful, yeah.
And there is a Acanthocarasica, which we think we've seen its burrows, but this is the first one we've seen today, first one in several dives.
Oh, there's two there.
And there's another one there, but that's different.
Oh, yeah.
That is once we get this one up and to the right is a different species of crustacean. That looks like Nephropsis or something similar to it.
So just a quick side note about that siphonophore we were just looking at.
Laser's
Dougal
clear.
has said that if we had to have collected it and then put our hand in the container, we would have suffered from a very strong sting. Very interesting.
Look at the transparent swimmerets under the abdomen of this little lobster. You see them
Max zoom.
rowing back and forth. Broad tail fan, large claws.
Does not have any eggs.
Nope.
We can say with surety.
So I think this is a Nephropsis, but I will.
And those are the species which we eat, right?
I think they are
like scampi.
Yes, I think they are. Maybe not the species, but I think they're related to some that are called Langostino. Let me just see if I can dig that one up.
Going into turbo mode.
I believe they normally burrow. Look at images here.
Interesting to see two different species that are so similar at first glance, so close to each other. So these are the blind lobsters, the Acanthocarasica. which we've seen, especially in our earlier dives, we saw loads and loads of.
And you can see the enormous claws, very long and slender, lined with all of these nasty spine-like teeth.
So Carolyn has just said, commenting on the geology, the carbonate here has much more sediment on top of it, but it is still layered and not so tilted here.
Let me see if my guess, I think there's a couple of Nephropsis in this part of the world. Look at that claw. And you do not want to have that close around your finger.
Video is clear.
And that is the classic posture that you normally see them in, with their sort of their elbows lifted and bent and the claws opened.
Looks like they're facing us on this side. You want to go full wide? Okay.
So this might be one of the Acanthogorgias that we were just looking at.
And Carolyn Ripple has commented that the carbonate rock here has more sediment on top than earlier. It's still layered and not so
Kill
tilted.
lasers.
Fabulous. Thank you, pilot.
There's another skate egg case that's only partly overgrown. Yeah. So there are 2, 4, 6, 8, 10, 12, 14, 15 species of Nephropsis.
Video is clear.
All told. And so there are several that it might be. I'm not sure which one. I don't think it was a culeata. If anyone on shore can weigh in, maybe Agassiz. See if I can find a picture of that.
Can we put the lasers on quickly just to see how big this colony is? Okay, so those lasers are 10 centimeters apart, so it's probably about 60 centimeters, 70 centimeters across. Thank you, pilot. You can turn them off now. Again, some more of these egg cases.
Yeah, this seems like the place for them, doesn't it? There's a cup coral off left, small Galathea down at the base of this fan, and 1, 2, 3, 4, 5, 6 maybe of those egg cases, and they mostly look overgrown.
Pushing in.
And this one is what? Is this Paramuricea?
I love how the egg cases look with the closed polyps on them. Like little nodule, like nubby little extensions. It looks like a cactus, it does.
I think this is more likely Paramuricea. Can we have a comment on this from shoreside? And if anybody knows the genus of Ophiuroid, again, it looks like, at least superficially, like Ophiocrius, but it's got all of these little papillae or short spines all over the arms that the other species did not have. So Tina says this is a plexaurid. And Asako says Paramuricea.
Very top, just looks like one of those egg cases or whatever that thing is, reversed. The left hand side. Now coming to center.
And if we could pan a little below right, there was a clot of branches right there. And that's interesting. You see how maybe there was a former egg case there with its little twine or something, but you see how those branches have grown together in a mass rather than their usual narrow branching pattern. So maybe that's
something's happening. I'm still trying to hunt for the spot.
Kinds of things
Seems
that
stable now.
Settle on these octocorals. And Andrea Quattrini and Les Watling have pointed out possibly Astroschema for the serpent star, but a different one, as I said. They agree from the one that we've seen before. Thanks, folks.
Fabulous. Thank you, pilots.
And that ophiuroid looked to have a slight purple cast to it. Different color, too. And there's the slope upward in the distance.
Not seeing too much as far as targets go right now. Nav, you want to put another 30-meter move?
Roger that. 15 minutes remaining on bottom.
Copy. 5 minutes remaining.
Bridge, ROV Nav.
So we have just under 15 minutes left on the seafloor during this dive at
Yes, can we get a ship move, please?
Basin.
30 meters, bearing 180,
Bob Carney had commented
0.2
earlier
knots.
that it was, he believes it was named after someone.
Who? What?
Good copy, Bridge. Thank you.
Ah, right. So folks on the chat and on the line, we're going to have our dive call at 4:10 Central Standard Time. So 10 minutes after we come off bottom, please.
Tina says that the Paramuricea has been
Looks like the
included
targets
in
are
the
back off port.
family Plexauridae. So she says she doesn't care anymore whether we call it a Plexaurid or Paramuricea.
Yeah, I just picked due south. Thank you, Ridge.
Yeah.
It looks like we've lost the hard bottom, although there's something out in the distance a bit to the right of
Yeah, any
center.
sonar targets?
Yeah, we can get that the distance from our left hand with tethers and go over to that second slanting around to make sure that it has to line up.
Ooh, can we have a quick zoom of this yellow coral right here, please? Right by the ophidiid fish. So this looks like a black coral,
Lasers are clear.
maybe.
I don't trust my eyes anymore.
Oh no!
No,
that
it's
is terrestrial plant matter.
Yep.
But it's a nice little bit of structure for this ophidiid to hang out by. It's its Christmas tree.
It's got him a tree.
Yeah, Tina's suggesting that looked like corn. corn
or
plants.
grass of some
also,
sort.
Yeah.
Your hardest return is back off port. Oh, okay.
And the bioturbation is widely scattered here. You see occasionally these almost amphitheater-like
Tilt up off the sandy bottom. I can give you a far shot. You can pan around, partially
that
zoomed.
have been dug up, not complete rings. Andrea said the fish we passed just a little bit ago was Monomitopus or Monomitopus. And what do we have here?
It's like a large closed anemone.
That might be a sea anemone contracted on a bit of hard substrate. Take a look. Yes, so that's a contracted sea anemone. The large one, there's a snail or three on the left, and then there are some half a dozen or so cup corals, solitary corals. I don't know the genus. There are quite a number of them. Need Steve Cairns around from the Smithsonian to tell us what those are. He did a monumental work back in 1979, the results of his PhD work. There are little colonial anemones there too. And this, I think, is what we've been calling a hormathiid, with those knobs on its on the outside of its column. When it expands, its oral disc and tentacles are bright orange, and it frequently assumes a Venus flytrap type posture.
Bridge, ROV Nav. Yeah, I was wondering if this is the heading you guys are thinking about for recovery, or if you're going to want to change.
Small encrusting sponge there. See how irregular and cavity-ridden this rock is, and there you can just make out there are
Roger that. He just came in, so I'll talk to him.
Bellid worms in their little mud tubes. There's one just above that sponge. And there's a beautiful view of that solitary coral. And those are brachiopods below it. Two brachiopods, and you can really see their expanded lophophores, their tentacular horseshoes for filter feeding. I have no idea what group they are in. There are several species that occur at this depth in the Gulf of Mexico that are known, but you can imagine that because these live on irregular hard substrates, they'd be very tough to dredge or trawl. Very neat.
Video's clear.
Brachiopods were hugely diverse and abundant during the Paleozoic era, before the Mesozoic.
It's time to start getting in orientation.
About
Copy that. Pilot, I'm going to start stowing.
Million years ago.
I'm pretty much lined up.
And then they declined
significantly,
and the bivalves
all right with that?
diversified in their place. But there are still a few hundred brachiopods alive in modern seas.
Dive Soup wants us to come up a few minutes early because it's going to take them longer than usual to get set up. You had a target something like
that's
that.
a
That patch.
burrowing anemone, a Ceryanthid.
That patch was in our sonar for a while.
See what else we see there.
Watch lead nav. Yeah, unfortunately, it's that time. We have to come off bottom. We're having a little bit rough of a sea state here, so it's going to take us a few minutes extra to get set up for recovery, so we have to pull off bottom a few minutes early. But it is that time to come off bottom.
So we are about to come off bottom. We're coming off a little early. I don't know if you heard that exchange.
That's where I was in.
The sea state has gotten a little rough, so they want to begin the ascent a bit early, give us a little more, get us to the surface a little more quickly. But I believe our...
All right, I'm marking you guys as off bottom.
Very good. Thank you. I believe our post-dive meeting will be at the same time. Let's see if I can remember when that
There's
is.
your structure. Over there.
Diva mentioned it. And I'm blocking. Imagine it'll be 10 past. Okay, 10 past the hour.
Copy.
All right, we'll talk to you then. Thank you all very, very much for participating.
Copy that. All right, I'm going to take
Bridge, RV nav. Yeah, I wanted to let you know that the vehicles are off bottom. We are on our way up. Pilot okay to change rotation? Bridge, you may change that now.
Copy that. Delta 16, I'm closing.
Bridge, RV nav.
Starting winch.
Yeah, if you could just kind of keep me updated with your heading changes and moves so that I can keep the pilots updated,
about
please.
30.
But I'm going to let you proceed as you and Dive Super
Copy.
came up with.
Yeah, I saw that earlier.
So I don't have manip cams over here, so I can't tell if that's on the monitor or the camera. It's the camera. Thank you. So that you guys know, Dive Soup's up there with the bridge. They're going to try a few different headings, figure out a ride. All of them will be southerly.
Copy.
I'll let you know if anything changes.
Do you want to slow down our winch?
Ship is still stopped. I'm just going to let them know when we're 15 minutes and they can call up when they're ready. I'll let Dive Super know when we're at 15.